and its auditorium is booked. सर से एक बार पूछना, it's LT2 और auditorium.
LT2 है ना?
नहीं, क्लास तो LT2 के लिए.
यहाँ पे सोशल डिस्टेंसिंग तो नहीं हो पाएगी.
नहीं, वो गूगल मीट पे ही होगा. मतलब
गूगल मीट पे ही होगा. तो वील कम हियर एंड.
नहीं वो तो है, बट वो हम वहाँ से लॉग इन नहीं कर सकते.
नहीं, अपने उससे भी कर सकते हैं.
हाँ, अपने उससे है ना,
अपने रूम से, किससे, घर से, कहीं से.
तो मतलब हम ऑर्गेनाइजर्स होंगे उस मीटिंग के लिए ना. ठीक है, तो मतलब प्रेजेंटेशन, स्लाइड, नोट, वो सब.
हाँ जी.
हाँ ठीक है.
नहीं, I didn't understand.
No, if we are logging in from here, usually the organizer starts the meeting. No? So that means then you have presentation rights and you're in charge. But if it's here and you're somewhere else, then technically you're also one of the loginees. Then how do you switch?
कौन सुना?
UG नीचे चले जाइए. हाँ, ये वाला. बहुत दिन बाद इस कमरे में आ रहे हैं. जरा ओपन करके देख लीजिए. आ रहा है ना?
हाँ जी.
ठीक.
हेलो, राम राम. कहाँ है कमल का? बात करिए बात. हेलो, राम राम. अच्छा कमल, ये बता, जैसे कि वो मैडम यहीं से क्लास करना चाह रही है, तो मैंने उसको तो कर दिया, क्या नाम है, मैं अपने वाला कैमरा वो खोल दूँ ना, माइक. ठीक है, बस और कुछ नहीं करना ना. हाँ, शेयर आ गया. ठीक है, वो खोल रखी है. हाँ, आ गई आ गई आ गई आ गई. नहीं नहीं, पीपीटी वो ओपन हो गई है. स्क्रीन में. अब उनसे कैसे पूछूंगा मैं? माइक भी ऑन है. एक मिनट होल्ड. हेलो, उसको म्यूट ऑल से हटाना पड़ेगा ना? म्यूट ऑल रहने दूं? अब किससे बोलूँ, 3-4 लोग आए हुए हैं इस पे, उस पे उनके माइक उस पे जाऊँ? नहीं नहीं, मैंने अभी जैसे ना उनकी स्क्रीन वो दिखाई दे रही है। हाँ कर दी। हाँ हाँ वो कर दी, स्क्रीन भी आ रही है। हाँ हाँ वो आ रहा है। अभी दिख तो नहीं रहा है उसमें। ना कैमरा नहीं दिखाई दे रहा अभी। हाँ। हेलो, कोई स्टूडेंट है अभी कोई? हेलो। वो तो जा ही रही है खैर। अच्छा ये जो ऊपर जो आ रखा है साइड से, वो उसको उसके नीचे कर दूँ, कहाँ कर दूँ उसको फिर मैं? नहीं, फुल स्क्रीन फिर कैसे दिखेगी फिर? और इसको नीचे कैसे करूँगा? भाई स्क्रीन पे तो आ चुकी है ये। मैं अपने उसमें डाल के देखूँ, फ़ोन में? हाँ हाँ हाँ। ठीक है ठीक है। ठीक है, आवाज़ भी जा ही रही होगी हमारी। ठीक है ठीक है। हाँ हाँ ठीक है। यार ये आ तो गई है, देखे तो एक बार इसमें। आ तो गई है ये, वो तू अपने फ़ोन से वो करिए बार, कैसे जोड़ दें वो?
आ तो गई।
अब वो दिखाई दे रही होगी ना वहाँ पे उनको?
एक मिनट रुको।
दिखाई दे रही ना यहाँ पे, देखने आ गई।
हाँ चल तो रही है।
चल रही है।
हाँ चल रही है चल रही है।
ठीक है, 9-10 बजे शुरू होगी ना?
यहाँ से कुछ नहीं करना है, आईडी डालनी है बस। अरे ऐसे नहीं होगा, उसमें ऐसे ना हो, ये ऐसे लो। नेट चल रहा है?
हाँ।
रुकना ही मत बोलना ऐसे। आया नहीं, कोई डॉक्टर नहीं आया?
मैडम स्लाइड तो दे के चली गई है।
ये लो चल रही है, ये आ रही स्लाइड।
होगी ना?
सुबह ज्वाइन कर ली, स्लाइड आ गई।
तो हमारा
फिर
ये ऑफ ही रहेगा ना एक मोबाइल
हाँ,
का?
हमें कुछ नहीं करना अभी, अपना आप करेंगे। प्रेजेंटर बना दिया?
वो कहाँ से बनेगा?
support about to go to meeting.
हेलो हेलो
हेलो ठीक है ओके स्पीकर ऑन कर दो ऑल म्यूट कर दो ऑल म्यूट यहाँ रहने दो स्पीकर ऑन है तुम्हारा हाँ ये बंद करो ना ये भी तो बंद है हेलो हाँ अब आ गई वीडियो हाँ, ना
तो ये आवाज़ गई क्या है
तुम्हारा स्पीकर ऑन है ना बस अब आ रही है अरे ये रहा ना ऑफिस से करो यार स्क्रीन डिस्प्ले ठीक है
पीपीटी देख लीजिए मेरा है
हाँ जी
एक पीपीटी देखना है
पीपीटी देखना है
इसके बाद होगा
वो तभी लग जाएगा उसके चेक करना है अभी
चेक भी कर लो एक बार
ठीक minimize कर।
कौन सोलर
यहाँ पे क्लिक करोगे यहाँ पे कितने लोग जुड़े हुए हैं
नहीं नहीं वो नहीं पहले पीपीटी देखना है
हाँ पहले ये
ये
देखो
एक ही होगा यही है हाँ
ओपन ओपन ओपन
यही
एक बार पावर पॉइंट पे खोल लो
इसको फुल कैसे होगा
पावर पॉइंट
ये नीचे कैसे हो सकता
ये होता है ये इस पे करो शायद ये वाला नहीं
है अभी तो बच्चे आ गए नहीं तो
इधर मिनिमाइज कर दो ना कट क्यों करो कट मत करो कैंसिल करो मिनिमाइज कर दो चलो
ठीक है
पावर हाँ इस पे करो इस पे हाँ एक बार
ठीक है
चला के देखो एक सेकेंड हाँ एक मिनट सारा नहीं आया क्या फिर से चलाना
ये ना आपके साथ स्लाइड है
हाँ चल रहा है ओके
हेलो सर
नमस्कार
ठीक है ये दे दीजिए
कट कर
निकालना
दो
है
सीधा
यहाँ से निकालना होगा
हाँ कट कर दो हाँ
आपने इसमें सेव कर लिया अब मुझे
हाँ
ये
कर दिया
चाहिए ना
कॉपी पेस्ट कर दिया
बस यार है ना देख अपने इस पे देखता हूँ
वो मिनिमाइज कर दिया तो निकाल दिया नहीं आ रहा ना
इस पौधे समझ देख अरे नहीं वो निकल इस पे
यही तो है और यही तो खोल रखा है एंड इनवाइट मीटिंग ऑन से हमारी ये है ना
नहीं नहीं वहाँ तो वो कर रखा है सेशन ऑन है हमारा
क्लास चल रही है
हाँ अरे सॉलिड खोलना है इसका देखो इसका देखता
ये है
हाँ
इसमें कौन कौन बच्चा अभी तो पता ही नहीं है रुको एक मिनट, यहाँ से उतार देना सर। 42 बच्चे जुड़े हुए हैं,
ये सारे जुड़े हुए हैं।
ये आगे चल रही मीटिंग।
ठीक है। ये मेरा करवा दो, इसमें ना एक बायोकेमिस्ट्री इंट्रोडक्शन करके है।
ये लो बायोकेमिस्ट्री इंट्रोडक्शन।
कौन सा है? BBB बायोकेमिस्ट्री इंट्रोडक्शन MBBS 2020।
इसी को कॉपी कर,
एक 4 करके है ना? हाँ,
ये 4 है सर।
खोलो एक, हाँ कॉपी कर दो। हेलो, गुड मॉर्निंग, मॉर्निंग।
ये सर।
हाँ, थोड़ा नीचे जाओ, और नीचे, और नीचे। हाँ, ये ठीक है, बिल्कुल।
जो भाई बच्चे आ रहे हैं ना,
नहीं जोड़ तो रही, पर जैसे वो लेक्चर कैसे होगा, लेक्चर का बता दो।
छोड़ दो ना, तो हो तो रहा है, कैसे होगा मतलब?
ऐसी है जैसे ये
छोड़ दो।
ये स्लाइड कैसे उनको दिख रही होगी, ये तो है।
दिख रही होगी, दिखाओ।
कहाँ दिखाऊँ?
फोटो मीटिंग पे जाओ ना, उस पर पता चलेगा ना, जरा उस पर, इस पर, इस पर जाओ, इस पर स्क्रीन। वो कहाँ से उतार दे सर? वो तो उतार देगा प्रेजेंटर वगैरह अपना। रुको एक मिनट, यहाँ से नहीं था वो। स्पीकर ऑन है, अनम्यूट ऑल। नहीं नहीं, चल रहा है सबका। म्यूट ऑल कर दोगे तो म्यूट हो
वो
जाएगा।
तो limit हो
अनम्यूट।
गया।
प्रैक्टिकल ओवरव्यू।
वो कहाँ से आ रहा है?
वो जब तो दिखा रहा था, अब क्यों नहीं दिखा रहा है?
जब तुम्हारे बैठे कमल को फ़ोन करो ना एक बारी, मेरे हिसाब से तो चल रहा है।
ये बच्चे तो जुड़ रहे हैं।
आ जाएगा कोई थोड़ी देर में। गए बिल्कुल तो बच्चे गए।
PPT has a name. Yes, sir. Yes.
ये आ गया ना, देख ऑर्गेनाइजर।
रुको, ऑर्गेनाइजर नहीं, प्रेजेंटर बनाना। ऑर्गेनाइजर बना दे तो।
इसी पे कर दूँ ना?
हाँ, ऐसे भी कर दो। वहीं से पढ़ा रहा हूँ ना अपने ऑफिस से।
यहीं से बढ़ाएंगे।
तो वो नहीं करना था ना, यहीं से पढ़ाएंगे। कैसे आया था, कहाँ से आया था? रुक जाओ एक मिनट।
मैं एक ऑर्गेनाइजर।
नहीं नहीं, ये इसमें मत करना।
वो तो कहते हैं छड़े की जरूरत ही नहीं आएगा, जो यहीं से क्लास करेंगे।
और उसकी तो ज़रूरत ही नहीं थी ना, तुमने कर दिया।
तो उससे वही वो तो वापस हो जाएगा।
कैसे होगा?
अभी तो स्लाइड शो कर रहे थे लोग क्या बोले इसको।
या तो बच्चे जो बता रहे हैं,
बच्चे तो जो है स्लाइड दिख रही नहीं दिख रही मुझे।
पूछो एक बारी।
हेलो,
हेलो तो करो, फिर उसको ये बता, बोलो बोलो।
हेलो,
तुम्हारे से होगा, तुम्हारा वो है ना प्रेजेंटर।
हेलो, स्लाइड दिखाई दे रही है आपको?
ये गूगल मीट के थ्रू है।
नहीं नहीं,
गो टू।
गो टू मीट।
पूछो किसी को, आवाज़ आ रही है, आवाज़ तो आ रही है।
हेलो, स्लाइड दिखाई दे रही है आपको?
फिर तो क्या करें? बोलो।
हेलो, स्लाइड दिखाई दे रहा है आपको?
वहाँ से ऑन करो वो स्पीकर।
फोन ऑफ करके इसको ऑन करो तो शायद आ जाए।
इसका इसका कोई मतलब नहीं। ये लोग
ठीक हुआ।
स्टूडेंट्स आ गए?
ऑनलाइन
ऑनलाइन है? जस्ट आस्क एनी देम वेदर इट इज स्लाइड्स विजिबल टू देम। बोला कि नहीं?
नहीं, अभी चेक करें।
ये ऑन है ना, इसलिए इको है।
इसका स्पीकर कहाँ गया?
इसको म्यूट कर दो। इसको म्यूट कर दो।
ये म्यूट है।
म्यूट है? फिर क्यों हो रहा है इको?
पता नहीं क्यों हुआ
सब स्टूडेंट्स नहीं है? अच्छा, कैमरा ऑन यहाँ से है कंट्रोल? सबका ऑन कर दो तो।
तो। हाँ अब बोलो। ये बोलोगे तभी इसमें
इससे
इससे नहीं।
हेलो,
कुछ स्लाइड दिखाई दे रही है स्टूडेंट्स को?
स्लाइड दिखाई दे रहा है आप लोगों को?
मना करो।
दोबारा से
ये लिंक नहीं हो रहा,
लिंक तो होगा।
साउंड तो जा रहा है, स्लाइड्स नहीं दिख रहा है।
स्क्रीन शेयर नहीं दिख रहा।
No, sir, we are not able to see the slide.
ओके ओके।
बट वी आर ऑडिबल टू यू।
हाँ, but without slides it will
हाँ.
not be possible.
ओके,
रिस्टार्ट कर
Yes, ma'am, we can
Fis-
see you, but we are not able to see.
ओके, वी आर जस्ट ट्राइंग, जस्ट वेट फॉर अ मिनट। ये फिर से स्टार्ट करके इसको बंद करके फिर से खोल दो। कहाँ पे है slide? शेयर होगा ना? इस पे देखिए, स्क्रीन ऑफ है ना? ये क्या है? शेयर स्क्रीन है? शेयर, ओके। अब स्लाइड खोलो, अब स्लाइड खोलो।
Yes, ma'am, now it is.
Okay, yeah. I just open this one. Is the slide visible?
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Okay, thank you.
तो अंकित सिक्योर है तो यहाँ बैठ के प्रेजेंट करना होगा।
हाँ जी सर, बैठ के।
अच्छा अच्छा।
वीडियो ऑफ है?
नहीं दिखाई दे रहा है।
दिखाई दे रहा है।
तो वीडियो भी ऑन है और स्लाइड शेयर भी है ना?
हाँ,
अच्छा, ये फुल स्क्रीन कर दो क्या?
हाँ,
फुल
सर।
स्क्रीन हो जाएगी सर।
फुल स्क्रीन कहाँ पे है इसका? ये वाला
हाँ।
कैमरा व्यूअर है।
सर यहाँ पे क्लिक करिए एंड यहाँ पे स्लाइड व्यू ऊपर ऊपर
एक सेकेंड, हाँ
ऊपर ये वाला।
ये वाला ना? हाँ।
हाँ,
हेलो, गुड मॉर्निंग, आर यू एबल टू सी एंड हियर अस?
Yes, sir.
Oh, good.
Yes, sir. Yes, sir.
Okay, welcome. हाँ, so we start
नहीं है ना?
now. So many of us, हाँ, क्या करूँ? इसको थोड़ा छोटा कर
थोड़ा
दो।
सा वो होगा।
Sir, we can't see you.
हाँ,
छोटा करना है सर इसको।
नहीं, हाँ,
We
you
can't see you.
you
सर का
can't see
कैमरा
us.
ऑन करो।
Okay,
See the slides, not you. We can't see you, only the slides are visible.
okay.
Sir,
So, well, fine. I think once the slide share is off, maybe we will try and make ourselves visible. I'm somehow unable to do both.
come out of the slideshow, then they can
We'll
see
come
you
out
then.
of the slideshow, then you can see us. Okay. So let us start with the slides. बेटा यहाँ
सर
पे
यहाँ क्लिक करिए
यहाँ हाँ okay.
इसको छोटा कैसे करें
Can you see the full slide, full screen, everything?
Yes, sir.
Okay, okay.
छोटा करिए
Yes, sir.
Okay, fine.
अपना क्लिक करके
So, well,
इसको नीचे कर सकते हैं जो ब्लैक वाला
doesn't I can manage with that. Let it be. अब हिलाओ नहीं. हाँ. Okay. So, welcome everybody. And it is really, while it is new for you people have been doing online classes for a long time, and so have we. But we still haven't yet got used to the idea of sitting in front of an empty lecture hall and talking into a computer. So somewhere the it's we'll try our best. Okay. So welcome to the fascinating world of medical biochemistry, and from the Department of Biochemistry, AIIMS New Delhi, a warm welcome to all of you. Now, you know, all of you have done a lot of biology for your entrance exams, for your class 12, and then you must be wondering why again a whole lot of biology is coming back to you under the guise of biochemistry and even physiology and anatomy, because you all of you are very eager to treat patients and work your learn new things and see how you can make the world better. The point is that whether normal or diseased, ultimately the disease process is a manifestation of abnormal biology, and this is something that you must learn and to think. And if you are in AIIMS New Delhi, then you have to also think that you as a clinician or as an academic medical person or as a clinical researcher, doesn't matter where you are, whether you are in a village or a PHC or a super specialty hospital, wherever in the world, you have to be a thinking doctor. Now, in order to be a thinking doctor, what is it that you need? You need a knowledge, you need a skill, and you need an attitude. The knowledge, of course, is the knowledge base, the hard drive of your brain, which will find you the sort of whatever the input to speak from, to think about. The skills is, a lot of skills are important for a doctor. A doctor is not only a thinking person, a doctor is also a doing person. So whether it is putting in a an endoscopy tube, whether it is doing a surgery, there is a lot of skill involved in the process. Even skill is involved in setting up a good IV line. So that is important. And then there is the attitude, which is essentially the way that you have to really be on your mental peak all the time, to think and to decide in this huge mass of evidence and information what is best for the patient. So this is something your biology, your biochemistry does help you in building the base. And also, we also would like to teach you a way of thinking logically before you reach to conclusions. So this slide I have shown earlier, but I'll just go through it. So what we will tell you in biochemistry is how modern biology shapes medical thought and practice, and also the molecular and cellular aspects of normal functioning. The biochemistry of disease, which includes molecular biology and cell biology. अरे slide off हो गया रे. Slide off.
प्रोजेक्टर पे कर लो.
हाँ प्रोजेक्टर पे कर लो फिर. Okay. Students, are you able to see? वो muted है ना, कह नहीं सकते ऐसा.
Sir.
Are you?
Yes.
Okay, okay, okay. ये सब हटा दो. Okay, fine, thank you. So and then we want to do investigations for the disease process, for diagnosis and for management and for molecular medicine. Okay, यार ये आगे से ऐसा मत किया करो, ठीक है? Okay, so they, just one second. मेरी स्क्रीन पे दे दो यार ये सब. Okay, so I will just give you, fine. Okay, let's start. Let's leave it as it is. ये स्टूडेंट्स के स्क्रीन में ये ऑब्स्ट्रक्शन आएगा?
उधर तो दिखाई दे रहा है
पूरा ये काला काला जो आ रहा है, ये भी आएगा क्या?
इसको फर्स्ट मिनिमाइज करके इसको ड्रैग करके फिर एप्लीकेशन ऑन करेंगे तो फिर इट विल बी हाई.
Okay, do that. Sorry, just wait a little bit.
कुछ भी नहीं दिख रहा है. पहले नहीं, ये आप स्लाइड पहले आप इसे यहाँ से निकालो. नेक्स्ट करिए, यहाँ से 1 मिनट.
Is that go-to meeting icon is showing up in your screen, or you are just seeing the PPTs?
The PPT.
So there is nothing.
Only PPT.
Okay, wonderful. Sir, they are not seeing it.
Okay, very good. Thank you. Yeah, there is a little bit of obstruction on the screen. चलो आ जाओ उस पे. हाँ. So now I thought earlier, you know, we used to have an interactive session and we used to ask people what is it to recount any recent events which are related to health and let us study the biology behind that recent event. And let us see how our knowledge of biology and biochemistry really helps us in understanding that recent event. But now I will just go through an example which all of you virtually would have encountered in your daily lives through a relative, through a friend, through somebody elderly or sometimes even someone young, about diabetes. So let us go through diabetes very briefly. Don't have to remember anything, don't have to, all this will be taught to you in detail, but just to see how what we teach you will impinge on the way you study and you perform your clinical functions. So diabetes is a syndrome which you know is characterized by hyperglycemia, high blood glucose, and this is caused by the absolute or relative deficiency of insulin. Don't remember this definition, not at all, not yet, but there is high blood glucose, insulin effect going on. So what do you do when somebody goes to the lab? What do they do? There is an emergency glucometer which you can use a finger prick and look at the blood glucose level. Now how does the machine look at the blood glucose level? This glucometer actually uses color chemical reactions to estimate the blood glucose level. So the chemical reaction, the reaction of the enzyme in that glucometer strip is proportionate, gives out a readout which is proportionate to the amount of glucose in the blood, and that is actually a reasonably accurate way of measuring the blood glucose. And that is sufficient for many purposes. Now, after that, what we have here in our laboratory medicine is a huge automated smart lab where we can do literally thousands and tens of thousands of parameters a day for the whole hospital. Now that requires really an automated chemistry, but the chemistry is the chemistry. The automation is in the machine which will accurately measure so many of your parameters. So then again, all of you know when you go, when somebody has to get their blood glucose tested, they go fasting and then they are given a little breakfast of not a little breakfast, a defined amount roughly, and then their blood pressure, glucose is measured after 2 hours. Now why? Why is it that we do all this? Because obviously it is common sense, but we will also know the biology behind it, the biochemistry behind it, the hormonal changes which go on, when you swallow some food, digest it, the glucose goes into your bloodstream. So why do the blood glucose levels behave the way they do? What are the endocrinological signals which ensure that the blood glucose levels which have peaked up after your uh, meal, they come down? So it is important to know this, and this will help you to understand the disease better and then to manage the disease better if you know the whole metabolic cascade that actually follows the assimilation of glucose in the body. And so we come into one thing that you will come in soon. I am giving a typical example, though you will study the biochemistry of carbohydrates, proteins, lipids, nucleic acids, so many other things. But just as an example, we look at in intermediary metabolism, what we say is that all these metabolisms are interlinked. So the metabolic pathways of proteins, carbohydrates, and lipids are all interlinked to each other. These are beautifully regulated. How are they regulated, and how does their dysregulation result in the disease? How do these all processes integrate together? I mean, you can't have a glucose molecule say that I am going to behave this way and a lipid molecule say I am going to behave this way. No, it doesn't happen. They are all integrated in a scheme of things which is very important to understand. And then the diseases which occur due to a defective metabolism. And then from a disease point of view, what do you look at? You look at the consequence of high blood glucose, and sometimes when a diabetes is given too much of insulin, the blood glucose drop precipitously. So the consequences of having too little glucose in your body as a consequence of either diabetes or some other cause. Then that diabetics also, they have an increased risk of heart attacks, of stroke, of kidney disease. Why does it happen? What is it that we can do to prevent it? Many diabetics who are uncontrolled have a phenomenon called diabetic ketoacidosis, where ketone bodies really go high up in their blood as a result of abnormal metabolism. This is potentially fatal, dangerous. You need to really look out for it, need to know why it happens, need to know how to diagnose it, need to know how to tackle it when it's there in front of you. And then again, a little bit of biochemistry again. What is insulin? Insulin is a protein. So you look upon insulin as a protein, and you can look, we will very soon you will be starting the module on proteins. So you will need to know that okay, these are a protein really is a string of amino acids. It's not just a simple string. It's a very defined string, and these amino acids have all side chains which have all sorts of unique structures, and they fold up and they interact to give you a function. So in biochemistry again, and also later on in pharmacology and other things, you go into this world of structure function relationships. How does the sequence determine the structure which determines the function? And many of this is caused by the interplay of different kinds of amino acids in this protein hormones. Of course, other molecules have their own structure function relationships. And this is a pictogram of a folded 3D structure of an insulin. There are two chains finally. Okay. Now comes, the first Nobel Prize on insulin was in 1923, when Banting and Best, as you probably know by now, found that pig insulin. So before that, people with diabetes, really there was no way you knew how to manage them. So from pig pancreas, you could isolate insulin and use it to treat diabetes. Now, as you can see here, one, that this is, not a very sustainable and easy way of getting insulin. Second thing is there are some minor differences in the amino acids of human and pig insulin. And while the pig insulin does function in your metabolism, there is sometimes the issue that antibodies will come up. So, in order to do that, now what we have today, what do we use most of the time? What we do is we use human recombinant insulin. That means the insulin, that means a product by a relatively recent and newfangled technology of recombinant DNA production is really being prescribed by any practitioner anywhere, whether in a small setup or in a big hospital, so on and so forth. So, and this E. coli bacterium, which is the workhorse of our recombinant DNA technology, because it is the most studied living organism in the world, is used for the mass production of recombinant human insulin by recombinant DNA technology. I'll not really go into that, but a little bit when we are talking of E. coli, we also want to talk about the commonality of biochemistry all over. Most of the metabolic pathways, whether E. coli, mouse or man, are very similar. Don't think that human beings are very unique in terms of biology all the time. They are. There are some very specific human specific features. But there is a lot of knowledge. In fact, the bulk of the knowledge that you are going to be taught is going to be very, very similar. There are very important human specific features that I don't want you to forget. But you know, the kind of, it also gives you an idea about the uniformity of life in general, that right from bacteria to human beings, for example, the genetic code is the same, the intermediary metabolism processes are the same. So this is something for you to ponder about and think. But coming back to our specifics, so the E. coli is used in the mass production of human insulin by recombinant DNA technology. The gene has been basically cloned from uh, human messenger RNA and then spliced into by a bacterium. The DNA of which is corresponding to the messenger RNA has been then spliced into a vector which is put in a bacterium. You all know about it by now. And then this produces the recombinant protein in liters in huge fermenters. These are industrial fermenters. So same thing is going to be used, similar stuff, more sophisticated, when you say that today because of COVID we will be able to give so many crores of vaccines in a short time to basically immunize the entire or a large majority of the human population. How is that possible? That is possible because of the advances of recombinant DNA technology. Mind you, we knew COVID, the first case of COVID was less than a year ago. Today, because of our knowledge of biology, which I don't see a distinction between biochemistry, biology, molecular biology, cell biology, microbiology, whatever it is, we know that it is a virus, we know the gene sequence, we have cloned different proteins, we have used this in order to test for drugs, we have used this cDNA, these proteins, to make a vaccine. Today we are making mRNA based vaccines, DNA based vaccines, protein based vaccines, whole particle vaccines, a vast variety of vaccines that has never been known in the history of the world. All this is possible because of these scientific advances. So the other thing that I said is now that the protein is made by the E. coli, the protein has to fold properly. Even that is a technology which of course most of you will not use, but this is the technology behind what you are going to use. So it's important and interesting to know a little bit about it. that a protein which is misfolded is non-functional, a protein which is properly folded is functional, and of course there are certain proteins which can be made in E. coli and they will become functional, and there are certain proteins which require human cells. Now why will that, you will be taught later, so don't worry, but what I am saying that the structure and folding of a protein is which you will be taught about is quite important. Coming back to another aspect of metabolism, which, because I am going into this because you will come quickly on into metabolism, that glycolysis is a very inefficient way of making ATP. So long time ago, Pasteur, who is the founder of all our biology and our biochemistry, it's interesting to learn that the first job that Pasteur had, that was actually that of an industrial biochemist. The French winemakers wanted to know why their wine was turning sour, and because of this he discovered anaerobic he has an idea of, not he didn't discover, but he got an idea that okay, this is the yeast which is forming or these are the microorganisms which are forming the ethanol, and when the conditions in which they used to form ethanol, and through that he discovered this whole uh, concept that gave rise to the germ theory of disease. So anyway, so when oxygen is pumped into a glycolytic reaction, so the rate of glycolysis obviously reduces because it becomes more efficient. Oxidative metabolism gives you more ATP, so you don't need to go through the pathway so fast, and this is the Pasteur effect. And in 19 early 19 something, uh, there was this German biochemist Otto Warburg. He found that cancer cells have a strange phenomenon called aerobic glycolysis, and you consume a lot of glucose. Of course, this is to make other products which you will learn later. But then basically cancer cells are glucose hungry. So can we use the glucose hunger of cancer cells for locating tumor metastasis? And then what we do is that there is from the second carbon to we will remove an oxygen and we will get a structure called 2-deoxyglucose, and that can be actually so that will enter the cells, and after and we can also substitute it by a positron emitting fluorine molecule. So fluorinated deoxyglucose, FDG, is a very interesting molecule. It will use the receptors to get into the cell, and then in the cell it will get stuck because you need an oxygen to proceed in the metabolism. So glucose hungry cancer cell will gather a huge amount or a comparatively large amount of FDG, which is fluorinated, and this is basically it will trap fluorine, and then the presence of that glucose of that fluorinated glucose can be detected by positron emission tomography of F18. Now interestingly, in our nuclear medicine department, we actually have the possibility of making and using this, which is a part of your department of nuclear medicine. So you see how these applications and the basic knowledge goes into the clinics. And this is a picture. Now in this picture, which has been this slide which has been given to me by Professor Bal of Nuclear Medicine AIIMS, you can see these black dots. You don't try to, but you will be taught about it. But the black dots are the regions where glucose is accumulating in the lungs. So what he has got is a thyroid cancer which has metastasized into the lungs. Now this thyroid, these metastases are not visible through your CT scan, they are not visible through your X-ray chest. So this is how, by metabolic labeling and by um, PET scan, you are able to actually diagnose a cancer metastasis which is not otherwise possible. And in this case, again, you are using your knowledge of biochemistry to really diagnose tumor metastasis. This PET scan today is very, is very commonly performed in order to do work up a patient of cancer. So you need to know whether the person is operable, not operable. If a person's cancer has spread all over the body, there's no point in operating that primary lesion. So this is the way. So you treat with chemotherapy if the person, in many cancers, I mean, you'll know the specifics later. So, but broad principle. So this is one use that you can get. Then, we are in the world of COVID vaccines, and all of you in the lay press also, there is immunity, some people are protected, some people are asymptomatic when they are affected by COVID, some people get COVID and they get worsened not because of the COVID but because of their body's reaction to the COVID virus. So, and they all, you may have heard of a thing called a cytokine storm. So, I will not just go into this at all, but just to tell you, just to highlight that we will be also teaching you the basics of immunology and cell biology related to immunology, to tell you about active and innate immunity, adaptive immunity, how we go into these processes that occur when there is an immune response. And recently a Nobel Prize in Physiology was given to 3 people, one of them, all of them for immunology. One of them, Ralph Steinman, he was awarded this Nobel Prize for focusing on dendritic cells in its adaptive immune response, and this is again related to immunotherapy of cancers. You may have read about immunotherapy, you may have read of CAR T cells, which is basically related to immunotherapy of cancers. So you see the various fundamental discoveries on DNA, whether it is structure, function, everything. And finally, most recently, you may have heard of this technique of genome editing. In 2020, Emmanuel Charpentier and Jennifer Doudna were awarded the Nobel Prize for the development of a method for genome editing, which is called CRISPR-Cas9. Now, what it does, it utilizes a method that the bacterium has evolved, just like your restriction enzymes are also bacterial enzymes, in order to prevent DNA from invading it. We all know the bacteria is a parasite. Is basically has a parasitic existence sometimes with our bodies, but the viruses and other so bacteria are also subject to DNA invasion, which will alter their genetic material, and in that process, how do they protect themselves? So one of them is this kind of enzyme system, and this has been used for doing gene knockouts. And this is a slide which was loaned to me by one of our faculty, Dr. Jayant, and you can see there are 3 different genes which have been knocked out, and the protein has vanished in the gels. So on the right, as you see, in most cases the protein has been knocked out, and the specific protein. So this is a technique that we are also using here. And so I'll just briefly go into what we do ourselves also. This is our department. I've shown this slide earlier, but I will go a little bit into somewhat more detail to tell you what we are doing. So I'll just briefly go through the faculty and their research interests. You see, I work on basically I work on glioma, I work on molecular oncology, I work on long non-coding RNA and the genetics of dyslexia. Dr. Chauhan works on cathepsin L and its role in malignancy. He also works on oral cancer, and he also looks upon the role of DNA binding proteins this, and in its role in cancer. Dr. Rajeshwari works on the conformation of RNA protein, DNA protein complexes in human disease. She works on unusual RNA, DNA structures in neurodegenerative disorders. She works on the developing biomarkers using proteins and nucleic acids, and also the functional role of DNA binding HMG proteins in cancers. Dr. Kalpana Luthra works on immunology. She is looking at the immunology of, specifically on the immunology of HIV, and she is looking at how the immune response of the body can protect itself from the virus, and also she has a lab which makes recombinant antibodies from single cell sorting in order to which are broadly neutralizing antibodies to essentially neutralize the virus. Dr. Partho works on nanotechnology. He looks at nanotechnology to see how this nanotechnology can be used to novel nanotechnology methods, a way of how the nanoparticle goes into the cell and is retained by the cell, and how this can be used in order to increase drug delivery and improve drug delivery. He is also using the CRISPR technology to look at the to study HIV and to see how essentially latent infections and also of HIV can be countered. Dr. Anna Sharma is looking at the pathogenesis of cervical cancer, and actually I can't see, yeah, and urothelial cancer, carcinomas, and she's also looking at the role of sheltering complexes and microRNA in multiple myeloma. She's also studying gamma delta T cells and dendritic cells in cancers. Professor Kunzang Chosdal is working on the role of FAT1 gene in human gliomagenesis and its regulation, and also looking at the Notch signaling under hypoxia in glioma. Dr. Sudeep Sen is looking at he is studying human stem cells, whether in bone healing or neural stem cells, and also looking at the way in a model of cerebral palsy how it can be studied, how human stem cells can be used to the modulation of human stem cells and their differentiation can be affected. Dr. Archana Singh is studying metabolism, and she's looking and firstly looking at the alterations in mitochondrial DNA and the structure of mitochondria in different cancers, and also she's looking at the quality and nutritional status of our population, and looking at the study of how metabolism can really help in different areas. Dr. Archana Singh is looking at the biochemical and genetic and immunological factors which go into the pathophysiology of tuberculosis. and also looking at the development of new adjuvant therapies in the area of pulmonary TB. Dr. Shubhradeep Karmakar is looking at human trophoblast biology, the role of a TET protein in acute myeloid leukemia, and looking at blood stem cell biology. Dr. Jayant is looking at the molecular analysis of leukemias in a great deal of depth. He was the person. Well, there are three, I think, four labs which have successfully been able to employ CRISPR technology in order to study their own scientific problems. So, then Dr. Pramod, Dr. Ashok Sharma is looking at epigenetics. He is looking at the port gene complex in uh, different cancers, and his lab is called the epigenetic lab. Dr. Pragya Acharya is looking at different genetic models to understand the pathogenesis of two interesting diseases and their biomarkers, malaria as well as acute on chronic liver failure. Dr. Rakhi Yadav is looking at cellular mechanisms contributing towards the chronic inflammatory states which actually occur during obesity and the overlapping metabolic and inflammatory mediators in the tissue, in the adipose tissue during obesity. Dr. Riaz is working on gene regulation in cancer, and he is looking at the molecular chaperones regulating cell cycle and its disruption in cancer. He is also looking at oncoproteins in the dysregulation of chaperone function. Dr. Pramod Gautam is modulating and targeting HSP70 in lymphoma. He is looking at the implications of nanoparticle peptide complexes in cancer therapy and targeting stem cells for immunotherapy, developing HSP-based diagnostic kit for cancer. And this is a new field that we have started. Next is Dr. Siddharth Kundu. He is a mathematical, he is MD biochemistry from here and then a PhD in computer sciences. So he has done he is doing mathematical modeling and simulations of complex biochemical systems, mathematical analysis of biochemical reactions and algorithms for the molecular biology and theoretical biophysics. So with this, we go into briefly what you are going to do, which will be explained just now by our faculty. So we'll be studying classical biochemistry, molecular and cell biology, immunology and clinical biochemistry. And as I said before, you will have didactic lectures, problem-based learnings, practicals, case discussions. Undergraduate, please feel free to come to our labs and work with us, or at least consult us from wherever you are just now. And with this, I hope I've been able to give you an idea of how our department is structured and how we do. Can we briefly switch on the video so that you can have a look and say hello to people, or do we have, or should we do the undergraduate? Undergraduate first. Let's do the undergraduate introduction first. So Pragya will now come in with her thing. Dr. Pragya Acharya and Dr. Siddharth Kundu. Thank you.
This has completely gone.
Look, I'm seeing it.
Okay.
Which one is it?
I can't see.
इसमें स्क्रीन नहीं आ रहा. Hi everyone, can you hear us?
Yes, ma'am. Yes.
Okay, so my name is Dr. Okay, great. Welcome to the Department of Biochemistry and welcome to AIIMS. So I am Dr. Pragya Acharya and along with me I have Dr. Siddharth Kundu and we will be your UG curriculum coordinators. You also have two additional coordinators for your practicals, Dr. Rakhi Yadav and Dr. Shubhradeep Karmakar. And the four of us will be your points of contact in case you have any doubts. Otherwise, also our entire department is open for any questions and queries you may have during your tenure here. So let's start with what all the practical aspects of your course structure. You just had a very nice overview of what biochemistry is and what what is its impact in biomedical scenario and research. But you will essentially start learning about biochemistry from the very basics. And this year, because of COVID, as you know, everything has been delayed and your entire coursework has been compressed into a shorter time frame than usual. As a result, both you and us, we have to work as a team in order to make your biochemistry education top notch. So we will just go over an overview of what you will cover and what is expected out of you, and if you have any further questions you can ask any one of us. So this is your UG curriculum for 2020-2021 and typically we have a mixture of coursework and practicals as a part of your curriculum, which includes lectures, case discussions, problem based learning and practicals. Now you know that your entire month of December will be will be a teaching in an online module, right? So you know that, right? Yes, no, maybe?
Yes, ma'am.
Good.
Yes, ma'am. Yes, ma'am.
So we will.
Yes, ma'am.
Okay, excellent, excellent. So essentially your biochemistry lectures are going to be on Mondays from 10 to 12 in the morning and 2 to 4 p.m. We expect 100% attendance, and also on Thursdays from 2 to 4 p.m. Okay, so on Mondays in the morning half from 10 to 12 10 a.m. to 12 noon you will have lectures, and in the afternoon you will have practicals. And your coordinators Dr. Rakhi and Dr. Shubhradeep will later tell you how exactly practicals are going to be conducted in this session. Okay. Now, as Professor Sinha just told you that there are various angles and aspects to biochemistry. What you will be starting with are basic biochemistry, with starting from cellular structure, subcellular compartments. Then you'll go on to study the building blocks of life, which are all the essential biomolecules which make cells and tissues what they are: protein, amino acids, carbohydrates, lipids, proteins, and their biochemistry, their functioning, their regulation. Then you'll come to, so this first term is going to have a major metabolism focus, because metabolic pathways form the basis for all endpoints in cellular biology and tissue biology, as well as regulation of homeostasis in your body. So sir gave you an example of diabetes, and so that's a very important going to be an important theme in the first term, and you'll understand the basic biochemical pathways which lead to diabetes, what are the basic regulation of carbohydrate metabolism, lipid metabolism, amino acid metabolism, lipoproteins, minerals, vitamins, and later on in the second term you'll also learn about immunology and immunological techniques, and at every point we will try to have a clinical correlation. Now, while we are going through all this material, we would recommend that you go and review your work on the very same day or within couple of days, because this is a really short session and your exams are scheduled in the first couple of weeks of March in 2021. So you won't have much time between the ending of the session and the beginning of your exams. So your preparation has to go along with the lectures. And I'd just like to reiterate that all of us are always available to solve any doubts or questions that you may have. So these are some guidelines for your books and references. Harper's biochemistry and Lippincott biochemistry are typically considered the basis on which you will do all your preparation, and you'll use Leninger's biochemistry and Streyer's biochemistry as references. For immunology, we recommend QB's immunology, but all your lecture slides will have learning objectives and material that needs to be covered, the absolutely essential material. So use your lecture slides as a guide, because these books are written when in a their coverage is very vast, and they go very in depth into each of the subjects, and you can use your lecture slides as a guide to understand what material to cover from each of these books. So for your practicals, for okay, here, yes, okay, this is the response. If you have a question, you can just quickly type in your chat box or ask. And anyway, we are always available. We are available throughout the session, so you can ask us. Great. So do we want to have an introduction with the videos? You okay? So now, okay, Dr. Anupama, who is a resident in our department, will give you an overview of what will happen in the practicals. You have a slide? Yes, sir.
Hey, there's a question. How can we contact you and other doctors?
Okay. Okay. Anupama will take that question. She'll give you contact information.
What is the syllabus for first semester?
You have been so you have been shared a kind of a curriculum or a schedule, and that essentially has all the topics that will be covered. And as I said, we'll have lectures. Our lecture slides will have all the topics that you need to cover, which you can use as a guide.
You are saying how we get the PPTs, so we can share.
Yeah, we can share, and they will be uploaded on the Saral platform also. At the end of the class, we typically share the PPTs.
Also, we can make a common WhatsApp.
You should make a common email ID or WhatsApp group, and we will share all our lecture slides with you. As far as contact points, if you Google AIIMS Department of Biochemistry, AIIMS New Delhi, you will get faculty profile for everyone, and everyone's email IDs are listed there. So you can easily access that, and we can also make an effort to put our email IDs or phone numbers on the slides, which we will share, so that they can have easy access. So that, yeah, and we are always there in the department. You can come in any time. Okay?
Is it necessary that we must get the books now? Yes.
Of course. Yes, it's very necessary. Please get your books, and you can even read in advance.
And some copies are there in the library.
And some copies are also there in the library.
But they are not here right now.
We can also download. Harper is available.
So a lot of these books are also available online, which you can download. Okay, so as that can be done. Harper is available online. Even Leninger, QB, PDF copies of these books are also available.
But not the latest version.
Latest versions we then have to buy.
It's sufficient for them. Yeah.
For the basic stuff.
Hello friends, a very warm welcome on behalf of all the biochemistry residents and faculty members once again. So I am SR Dr. Anupama, and I am here to give an overview of biochemistry practicals. So the objective of having biochemistry practicals is that by the end of this whole session you should be able to demonstrate knowledge and understanding on the principle of various conventional and specialized laboratory investigations and instrumentation, and you should be able to make conventional techniques or instruments to perform biochemical analysis relevant to clinical screening and diagnosis, and you should be able to analyze and interpret investigative data and demonstrate the skills of solving clinical problems and decision making. So we have made a biochemistry manual for you all, and it is with the effort of all the residents and faculty members, and it has in details about all the practicals, and it will be very helpful to you. So you will be getting the manual when you will be coming here, attending your lectures and practicals, but we will be giving you the soft copy of this manual, so you can go through it and have a look, and it will be helpful to understand the practicals better. So practicals will be held in a practical laboratory. We have a very good, very big laboratory, and it can accommodate all of you. We have arrangement for around 120 students. But before every practical that will be held every Thursday, 2 p.m. onwards.
That is during
When you come, not for this December, but when traditionally we have it every Thursday, 2 p.m. onwards.
Monday को बोलो दिसंबर में मंडे को आफ्टरनून में और उसके बाद जनवरी से थर्ड.
In December you have it Monday 2 p.m. onwards, and from January the schedule will change, and the traditional schedule will be followed, in which you will have the practicals every Thursday 2 p.m. onwards. So this practical lab is in room number 4005, fourth floor, convergence block. But you don't go there directly. First you come to LT2, which is in academic block, for a practical briefing. So the residents give you briefing about the practical, what you will be doing, overview of there is a sort of lecture about the practical in the LT2, and then we move towards the practical lab, and there you get an hands-on experience. So this is the exam pattern that we used to follow. So as you can see, there is equal importance of your theory classes and practicals. They have same marks, they have equal weightage with respect to marks, but because your session is short this time, this year you will be having one exam less, but the total will be same. Theory and practical marks will be same.
Can you tell them tentative time for first mid-sem?
It's not there.
It's in March
1st to March 15th.
So first exam will be in March 15.
March 1st to March 15th.
March 1st to March 15th you will have your first exam. First mid-semester exam. So very soon you will be having your exams. Your session has just started. So this is the breakdown of your practical examination. You get two practicals in your exam. One is a major practical, which takes which is a bit longer, and one is minor practical, which is a bit shorter. And then there is a bench viva related to what you have done in your practicals, in your major and minor practicals. Then grand viva is taken by the faculty members. Okay, it is even the theory is asked in that. Then you have this MCQ SAQ exam, which is part of the practical exam. In this you will be asked question related to your practicals only. So you have the MCQ SAQ in theory exam also and in practical exam also. Then you have spotters and OSPE. So regarding the spotters and OSPE, we will be explaining you again when we will have your when you will have your instrumentation classes, that is in the coming weeks. So we will be giving you examples also what actually these spotters and OSPI means. I think spotters you must be knowing, because you must be you are given something to identify and you are asked questions on that. That are spotters. OSPI is something you are asked to demonstrate. Objectively structured practical examination. And then the total marks are 100, but then like this previous slide says, you are then the marks are taken out of 25. They are so finally the practical examination and the theory examination, they are of equal weightage. So once again, welcome all of you to our department, biochemistry department, and we are really eager to see you here. And till then, let's have this December month, let's just have these online classes, okay? And you have this query that you want to get in touch with us directly. So I will type in my phone number here. You can have my phone number. Hello? No, they can read it from here.
You're going to tell them, no? Briefly tell them what all practical you are going to cover and how it is possible to do online.
So this is a phone number, you can have this, and I would request the CR. Do you have a CR, class representative? Have you decided on the class representative yet?
They're saying, ma'am, please turn on your camera. That's not possible.
So who is the CR? Nitin Singhal. Hi Nitin. So I request you to get phone numbers of all the students. So what we have for the previous batch, we made a WhatsApp group. So that was very helpful. We have few residents there, few biochemistry residents there, and all the students. So it becomes very easy in case they have any problem, logging in the class or anything, and we can share links and everything there. You have a WhatsApp group, so you can share that with us. So I have given. So that is my phone number. So you can add me in the WhatsApp group, and I will add the other residents also. So any query, you can talk to us.
Mention your name also.
And I am Dr. Anupama, senior resident. So anything else you want to ask?
Nice introduction. Thank you. Anupama, can you tell them they have a lecture now and then they have to come back in the afternoon at 2 p.m. for the have a lecture now.
Okay, guys, you have a lecture now, and then for the practical you have to come back again at 2 p.m. Okay, so we will brief you in detail in practicals also. Okay, thank you.
So, black.
If you're going to take a lecture, you can switch back here so you can see. It's very hard to look at that while sitting.
Dual, no? Dual also.
Yeah, we'll call that back.
ये तो projector वाला. This is the projector.
This is the projector.
This is the projector.
If you open, then only screen will come.
Only screen will come. You want to open it?
Yeah, I just put my PPT.
Oh, okay.
But this is not this two, right?
Yeah. So this is the projector.
Because seeing that is very hard.
You know, no, it's very difficult. Now say anything.
I'll just see from the screen and get it. No problem, ma'am. Thank you, ma'am. Thanks, everyone. हाँ? नहीं निकाला ना फिर सारा गायब हो गया.
There's everything. He doesn't have anything on the screen. So there's nothing here and nothing here.
For the next lecture, we should.
Wait, where's the lecture process?
Yeah, I think now it's okay. Wonderful.
Coming there? No.
Yeah, it's not coming there. I'll just see from there. The students see that's important.
Hello.
Hello. Hi. Thank you. Thanks, Siddharth. Hello. Good morning. Can you see me? Oh, good morning. Yeah, wonderful. Just 2 minutes of break. Just stretch yourselves because you have been hearing so much of us talking around. So just 2 minutes of stretch. I'm still sitting over here, and after 2 minutes, you are going to start with the first class for the today. Okay? The cell structure and the function and the subcellular organelles. So we're going to start with that. Just stretch out for 2 minutes. I'm right over here only. Yes.
Sir, please record this lecture if it is not being recorded right now.
Is it recorded?
We can record it. I think you'll have to ask the LT.
So other thing is that the moment the class is done, I am going to send off this PPT to your WhatsApp group. So either way, you will have this PPT with you. But then you won't be able to visualize. Hear what I am saying. Just I'm trying to figure out if it could be recorded. Hold on, let me find out. The guy is there.
He's coming. I've called him.
So far, how you are feeling joining AIIMS? Is it exciting?
Yes, sir, it's really exciting and really nice.
Wonderful. Actually, we kind of missing you all guys because this is a very unusual year that everything is virtual. I had never done in my life that the entire classroom is free. There is not a single person. I'm just talking. If someone see me, they will think that this guy is a crazy. Whom he is talking to? So it's like that a very weird scenario where there is an empty class and we are just talking to the computer. So either way, this is another experience, a lifetime experience for all of us. Okay, so if everyone is around, then let's start with the first class today. Okay, is my slides PPT is visible to you very clear? Just give me once again.
Yes.
Okay, excellent. Yes, so we are just arranging to record. ये record हो रहा है?
हाँ record हो रहा है sir.
It's getting recorded, so you will have the copy of the record. Not a problem. Okay. So again, today's class I'm going to, put it very easy. It's just think it that it is an extension of your high school, whatever you have studied, and it's also an extension of, pretty much what you have been doing for your, med school entry, AIIMS and entrance preparation. So it's very basic, very fundamental, but at the same time very important. So we can't ignore it. But it's very light. I mean, so take it easy, and rest of the biochemistry is kindly slowly we are going to go into complex avenues. But today is very simple. Okay, so these are the lecture objectives. Over here I'm just going to see if this could be minimized or not. Let me see. Okay, I don't need that. No, no, no. One minute. Let me. I'm just trying to play around with this one because this is kind of blocking my slides. Okay. Hopefully, I think that's the best you could do. So you may not be able to see me. That's fine. I'm just kind of walking close to the slides and then actually explaining to you. And if I'm not audible, just kind of stop me in between and tell that I'm not audible. Okay. So the lecture objectives for today's class is we are going. So our head has actually fairly given you a good idea about exactly what you're actually dealing with. So we are not going back again and repeating the same thing. So in today's class we are talking about the cell, the basic structure of the cell, and the subcellular compartments within the cell, and the different organelles within the cells, right? So each one of these subcellular compartments are very important because as you're going through into the lecture you appreciate that individual components of the cell has their own individual functions, and if those functions are compromised or they are not been done properly, or if there is a disruption of the functions, then you will have a very serious pathology associated with those disruption of the functions. And therefore it makes it even more important that we do understand that what are the cellular components and what are the function of the cellular components, right? So that is actually the objectives for the today's class, and these objectives actually been written in those points that how exactly or what are the different components of the cells. Now over here actually something is also mentioned about the prokaryotic cells. So we are not going to talk anything about prokaryotic system, just to have one slide to tell you that these are the prokaryotic cells and these are the eukaryotic cells. But apart from that, the whole of the class is purely on eukaryotic cells. So we are not going any further detail about the prokaryotic or the bacterial systems, right? And the next is that we are going to study that the relationships of these subcellular compartments and the subcellular organelles in relation to each others. So in other words, I'll just give you an example, like if I'm studying mitochondria, but if I'm not studying mitochondria in relation to say lysosomes or in relation to the nucleus, then I'm not going to see the whole picture in total. So for studying, it's okay to study them in isolations and separate, but once you are trying to study the biochemistry, these are all very linked with each other, because as you're going to study, you will see that the cellular energy actually been produced in the mitochondria, but then the fuels of those actually been produced in the cytoplasm, and then they have been shuttled from the cytoplasm to the mitochondria. So this kind of a crosstalk between the different intracellular compartments and intracellular organelles is very fundamental for the living cell. Without that, it's not going to survive. But we are going to study each one of them separately. But remember again that they are all integrated together. Okay, so that's the cells are the building blocks of the life. Pretty much all of these things. So I'm going to go quite fast because this is very trivial, which most of you know by now, that the cells are the smallest living part of the organisms. The cell has the capacity to reproduce, use energy, adapt, response to the environment. Now this is something, again, I just wanted to have a word of caution, is that even though we are talking in this first class things in a very casual ways, but think about that these are really not casual in the real world. Why? Because when you say that reproduce, this means that the cells, one cell can actually form the daughter cells, right? It can actually multiply, it can undergo cell divisions. So that's a whole lot of interesting biology over there, that how the DNA is replicated, how the cells undergo cytokinesis, how the two cells are actually formed, and so forth. Because this is also very fundamental in understanding the different pathological conditions, like in cancers where you have over multiplications of the cells, or in neurodegenerative disorders where there is an under multiplications and then the cells actually dies. Then use energies. This is again very fundamental in understanding and correlating with the metabolisms and other things, how the cells can efficiently use the energies. Adapt and response to the environment. Again, this is fundamentally a very large domain of discussions itself. Each of these points can be taken into 2 hours of lectures, because you know how the cell responds. For responding to the cells, to the environment, the cells need to have on the cell surface something called the receptors, and exactly how the receptors transmit the signals back within the core of the cells by something called the second messenger and the third messenger systems, and eventually how they get transmitted to the nucleus by a nuclear signal transmission process, and then how the nucleus actually responds to these. So these are so many different steps. That's what I say, that these are worth discussing into couple of 1 hour, 2 hour lectures. But we are just trying to keep today very simplified, but underlying this simplified version there are undercurrents which are quite complex but very important. And as and when you are going into your biochemistry sessions for the entire semester, you will understand that each of these faculty is going to concentrate on those individual domains like reproductions, like cell divisions, replications, cell metabolisms, and so forth, and they are going to expand it further. So it is a very superficial today, but nevertheless we are going to have the flavors of this in a more detail once we are going through this rest of the semesters. And then the third point is the cells cannot be seen through the naked eyes. Well, we all know that, appreciate that, right? That's the reason we have something called the microscopes through which you actually look into it and then actually visualize the cells. But having said that, different cells comes in the different dimensions. Some cells are very small, very, very tiny, and some cells are actually larger. So I will have a slide which is going to show you the spectrum of the different sizes of the cells and the other things in comparison to that. The types of the cells, as I told before, that there are different types of the cells. So even in a eukaryotic multicellular organisms like us, we have so many different varieties of the cells because we have different tissues, right? And each of the tissues has their own cellular natures. Like muscle cell is different from cardiomyocytes, which is different from hepatocytes, the liver cells, which is different from the blood cells, which is different from the neuronal cells. So there is so many cells, but at the same time there is so much heterogeneity within the cell. Each of the cells, they look differently, the functions are different, but again at the level of the entire organisms, the entire heterogeneity converges to an uniform homogeneity. So as an individual, you perform our functions together, right? So everything is actually been integrated together, but at the core of it, there is lot of heterogeneity because different cells has different functions and different responsibilities. Now looking back into the history of this one, back in 1839, 1840s, 1839, the modern cell theory was actually kind of developed by two great people, Linden and Schwann, and they kind of, at those time, the science was pretty much under the very early stages of development. So they came with the 6 different postulates of the cell theory, which is remarkable and fantastic. If you think about 2020, 21, and then think about 1839, 1840s, back then where technology was not there, the scientist community who was working is very small. So making those conclusions are phenomenal. So they independently arrived to these conclusions that all living organisms are made of cells. Fantastic, right? Today we know that this has stood the test of time after so many years. Then they said that the cell is structural and functional entity of living things. Again, wonderful, because they understand that these are the living cells, and that actually what makes up an organisms. So wonderful postulates, and again this is still stood the test of the time. So this is awesome. The third point, actually I want to spend a little more time, that all cells comes from a pre-existing cell. Think about 1840s. At the time, the entire ideology and the political environment, this kind of more related to religious beliefs and all these kind of things. So telling at that time that look, has to be pre-existing cells that gives rise to the daughter cells is an absolutely bold statement, because people at the time was thinking about that God has created and de novo you can synthesize in cells, something like out of nowhere in the magic you could actually create a cell. But then they said that look, that's not wrong, that's not right, that's fundamentally wrong. You need to have a cell that's going to give rise to progenitor cells, and that's going to give rise to the further is daughter cells. So there's a lot of oppositions, a lot of criticisms, but nevertheless this is a fantastic hypothesis, very bold hypothesis back in 1839, to talk about the cells that they need another cell to replicate, another cell to start with, and not being created out of magic from nowhere is just remarkable. Now, point number 4 is that cells contains hereditary informations which is passed on from cell to cell. Again, if you think at 1840s, we really have no clue about hereditary. See, back in 1865, Mendel is the first person who actually did, crosses of those famous pea experiments, taking about 30, 40, 60,000 pea plants and then crossing them and then finding out that something called trait is been transmitted from parents to the offsprings. But this is about 40 years before that. So at that time, they don't have any clue about something called genes or hereditary or all these kind of things. But even then, on the basis of the observations that a blood cell forms a blood cell, a muscle cell forms a muscle cell, a hepatocyte cell, liver cell actually forms a liver cell, makes them to believe that possibly this information is retained through the generations when the cells divides. And therefore, there is something called hereditary, something called inheritance, which is actually been transmitted. Again, they are very sketchy, but nevertheless, this is remarkable, thinking that we were totally clueless about genes or anything back then. It is almost like 30, 40 years even before Mendel did anything with the genes or anything. And the 6th point is that all energy flow, metabolisms, and the biochemistry of the life actually occurs within the cell. Again, remarkable point, because there is nothing which is actually been outside this. The cell is the store of the energy. It's like a Duracell. It produces its own energy as a result of these chemical reactions, and eventually, that is the one that actually keeps the cell going. Now, this is the postulates of the modern cell theory, and we know for today, even in 2021, that fundamentally most of these are absolutely right. Okay, so this is something which is correct, but then I wanted to put your focus on the point number 5, which is not completely right, because point number 5 talks about that all cells are basically made up of the same chemical compositions. Now we know that each cell is actually being tuned into a different way, right? So the neuronal cells, the chemistry of a nerve cell is different from the chemistry of a blood cell, is different from the chemistry of a pancreatic cells, and is different from the chemistry of, say, of a kidney cells. So they are possibly fundamentally many things are similar, but they are not identical. So each cell is actually a universe within itself, because it has its own sets of chemical reactions which are fine tuned to serve the purpose of that particular cell and that particular tissues of which the cells are composed of. So point number 5 is a little thing which we need to be kind of skeptical about the cell theory, but then these guys did something in 1850s and 1840s, so we should give a big hands to them for making up this foundation theory. There is something called cytology, which is a real subject in pathology where people study the cell, and that actually gives fundamentally lot of information about the disease process. Today, if someone have a disease and it goes, the biopsy of the slides goes to the pathologist, then the cytopathologist looks into the slides under microscope and then say that, okay, this cells looks dysplastic, this cells looks necrotic, this cells looks highly mitotic, this cells look a little bit of apoptotic. So just by looking into the morphology of the cells under the microscope, the cytopathologist can tell a whole lot about the disease, that okay, this is the cell or this is the core of this entire tissue where there is hard cancerous cells are possibly over there, because you see a lot of proliferative cells over there. Okay, this is the core of the tissue where we see lot of cells which are dysplastic with the different shapes. So possibly these are precancerous. They're going to go into cancer after a couple of days or weeks. So cytology is a fantastic, strong, powerful field of pathology just by looking into the cell. So that gives more importance for the today's class, that understanding the cell, the fundamentals of the cells is so crucial in understanding the organism. So don't think that this is a separate entity. This is a part of the building block of what you are going to study in the biochemistry in the later on, and you are going to carry on in your entire medical professions. Now this is the slide I was talking about, gives a whole range of the dimensions of the different stuffs within the cells, right? So we have something at a very small micro levels to a thing which is pretty much large, you could actually see through the eyes. Again, this is a slide for you, just spend your time and understand the different spectrum of the sizes. Just I wanted to explain to you one small thing, and then you are going to go from this slide, is that the different sizes of the components we could visualize with the help of a parallel things which is happening not in biology but in physics. The physicists actually been studying optics, Newton was pioneer in studying a lot of different fantastic principles of optics, and then from that development of the physics, people started building the microscopes, and as and when microscope was developed, then this has actually enriched the biology. So all the science what we study today is actually an integration and a crosstalk of all the different disciplines: mathematics, science, physics, chemistry, all assembled together, and each one of them are actually been enriching the rest of them. So they are not separate, they are not different, they are just kind of flow from one to another one. If there had been no development in optics, no development in physics, forget about that. We have been not able to even see the first cell, because we have to use the principle of optics to see the cell. So this kind of things goes hand in hand. I'm just trying to put a focus on this one, that the things which are very microscopic over here, you could see that we need an electron microscope to see them, because they are so tiny that even your best compound microscope or the lab microscope cannot suffice, and therefore you need to go to a very powerful, where you beam them through an electron rays, and that's known as electron microscopy. You'll be very fortunate to have electron microscopy at AIIMS. So at AIIMS, in the Department of Anatomy, we have an electron microscopy facility, and there you have fantastic images of these subcellular organelles within the cells. So I have also put a couple of EM images. EM is electron microscopy images in my slides. We're going to show them. So this is the slide that actually talks about the different dimensions of the things within the cells. Again, this is for the importance. You don't have to kind of mug up those numbers. This is just an overview. So just sit back and relax. We don't have to be very particular about the size. Just an idea about the different ranges of the dimensions. This is the cells: prokaryotic cells, eukaryotic cells. Again, we are not going to talk about the eukaryotic cells, the bacterial cells, because this is in cell wall, and then we have the eukaryotic cells, which is in cell membrane. But nevertheless, I mean, the basic building pattern is very similar, because they had the same common evolutionary backbone maybe. But after that, there is a lot of divergence, and the eukaryotic thing has evolved in a different directions, and the prokaryotic things has evolved in a different directions. So this is the prokaryotic thing. Again, as I said, you will be studying a detail in microbiology. Maybe you have been knowing way more than what is given in the slides. So we are not just going into that. I'm quickly passing on these slides, telling that this is bacteria, and then this is the different components of this prokaryotic cell. Okay, so now we are going to see a sagittal section of an eukaryotic cell. And if you take an eukaryotic cell and kind of slice it on the slice of it, like something like you take a slice of butter, then you would see, and just if you could peep within the cell, as if you had actually been looking through a window in the attic, and then you would see that there are different components of the cells. We have the nucleuses, we have the endoplasmic reticulum, we have the Golgi complexes, we have the cytoplasm, mitochondria. So entire cell looks like an aquarium. So if you have visit an aquarium, say a very big aquarium, what is there, something in the Disney park and other places in California, very gigantic aquarium. If you just see through this aquarium, you'll see all the different fish, which is actually been floating in these aquariums, and different varieties, different species of the fish. That is something exactly what you're seeing over here. It's looking like an aquarium with different complexities within the cells. I'm going to talk about each of them very brief, but in the subsequent slides. Now this is something which is cell membrane. The next slide is the cell membrane is the outer coating of the cells, right? Outer covering of the cells. Again, I'm not going to talk too much detail about it, because our next class, which is on 10th of December, Thursday, we have an entire dedicated 2 hours of lecture for the cell membrane and the transport across the cell membrane. So I'm just kind of going very superficially about it, but then this is also something very important, which is going to be, important not only just for your understanding of biochemistry, but also for your future interpretation of the disease. Now this is the cell membrane, and then we have a very good theory about the cell membrane, which is known as the lipid bilayer theory. And in this lipid bilayer theory, it says that the cell membrane is actually a sandwich of 2 layers of the lipids, and it's something like an ocean of the lipids within which there are the islands of the proteins. So this is something like that. This is the ocean of the lipids, and these are the proteins within them, something like in floating islands. So that's the cell membrane for today's class, not more than that, because we have a dedicated thing over there. But something which has been very interesting in this image is that something which has been popping out from the surface of those proteins. What are those? Those are glycoproteins. Those are the sugar residues which has been attached on the top of the proteins. Why they're important? They're very important. We are going to talk about them. There will be separate class. People are going to take an entire lecture on that what are glycoproteins. But understand that these glycoproteins are very important, because they help in the cell recognition. So when the two cells try to interact with each other, these glycoproteins are the ones which are the sensors, the radars. They are going to sense that, and they're going to know that, okay, how these two cells are going to communicate. Many times without the glycoproteins, the proteins are non-functional. So they need these glycoproteins. So very important substances on the cell membranes. Okay. So that's the plasma membranes. again I'm going to talk about enzymes are hooked to the plasma membrane. A lot of receptors are there on the plasma membrane. So plasma membrane is actually wonderful place for us to start our basic biochemistry. That's what we are going to do in our next class. Okay. Now, once we cross the plasma membrane, what you see is the cell soup, and what is known as, that's known as the cytoplasm. So the entire cytoplasm is actually a soup of within the cell. So the cell is actually in bag, and this is the soup within the bag. Now this cytoplasm is again a very important component, because that's where hundreds of chemical reactions are taking place. If you go into the cytoplasm, think it as a swimming pool, and if you plunge into the swimming pool, you are going to see that thousands of chemical reactions are actually been boiling in the cytoplasms. Boiling is not in the sense of heating and boiling. I'm just trying to put you an influence about that hundreds of reactions are taking place in the different pockets of the cytoplasm, and we have these different reactions like glycolysis, glycogenolysis, pentose phosphate pathways, polysaccharides, complex lipid biosynthesis, fatty acid synthesis. All of these processes actually takes place within the cytoplasm. Another remarkable thing about cytoplasm is that cytoplasm environment is reducing in its nature. And because of this reducing nature, it's not oxidizing, it's reducing in its nature. Because of this reducing nature, many chemical reactions can take place only in the cytoplasms, not in the other organelles. But having said that, cytoplasm is not the only most reducing environment in the cells. The most reducing environment in the cell is within the mitochondria. And the nucleus is also sufficiently reducing. But cytoplasm is also sufficiently reducing, because there is production of some reducing equivalents in the form of NADH. We are going to study that again detail in your metabolism classes, but those NADH which is produced as a result of these metabolic reactions, they make the entire cytoplasm pretty reducing, and we need that reducing environment to foster further chemical reactions. So in that way, it's a very big chemistry labs, as if you think it in that way, the entire cytoplasm is. The other important thing, which is why this class, is that you might ask this question, that why do we need to have the different organelles within the cells? What is the reason? What is the that we don't have a system, something like in bacteria, where you have everything all mixed like a soup? Why you need to have an endoplasmic reticulums and Golgi complexes, and then the lysosomes and peroxisomes and all this so forth, the different components of the cells? What is the reason for that? Well, the reason for that is that most of you are possibly aware of. I'm just telling you that the reason is because each of these different organelles within the cells are actually the factories of chemical reactions, and therefore we need them to be enclosed in their own privacy. We don't want the privacy to be disrupted. If you take the mitochondria and just pinch it open, immediately the mitochondria lyses and it becomes dead. It doesn't serve any purpose. Mitochondria is gone forever. So in order to maintain this complex mitochondrial reactions like electron transport chains and fatty acid metabolisms and so forth, we need the mitochondria to be enclosed within its own privacy, similar to that of the lysosomes. The lysosomes contains all hydrolytic enzymes. If you pinch open the lysosomes, it's going to destroy and kill the entire cell. So you need to retain the environment of those individual organelles, because each of these individual organelles are actually streaming with their own chemical reactions, and we don't want them to be mixing up with the chemical reaction which is happening in the other organelles, and therefore each one of them is actually in compartments. It's something like a big villa where you have individual rooms, and each of the individual rooms have their own doors, and people staying in those rooms have their own privacy once that closes their doors and the windows. But having said that, each one of them can also open the door and communicate with the neighbors in the next door in the entire big villa. So it is some same thing like that they had the privacy, but at the same time there is also a fantastic crosstalk between the different organelle systems. How wonderful the crosstalk is, we're going to see in a couple of these slides later on. But you'd see that the entire system is actually built up in this way, that we have the privacy and we also have the crosstalks, so that the information can be shared. Okay, so this is a journal. It's your wish to read this or not. I'm not forcing on you, but this is very way to go with and develop and cultivate your scientific reading habits, like redox compartmentalizations within the eukaryotic cells. If you read this one, you would see that how nicely they explain that the different compartments are actually being kind of engaged in their own chemical reactions, and what are the different reducing environment within them, how much is the acidity within each of these environments and so forth, which tells us that all of these components are actually different. Okay, so that's the cell, that's the nucleus, and these are the different components of the cells. We are going to go one by one into them. So this is eukaryotic cells full of membrane enclosed compartments. That's what I told just now. These separate compartments are compatible with the chemical and the physical conditions, and that the way the proteins can be synthesized and hydrolyzed within a single cell. So within a single cell, you know for sure that there are some places where proteins are synthesized. Within the same cell, there are also the places where proteins are degraded. So we have the lysosomes within the cells, and then we have the endoplasmic reticulum where the proteins are synthesized. How it is possible if they are all together? So we need to compartmentalize them and make them into separate enclosures so that we don't jumble up and make them chaotic, right? So that's the whole purpose of this one. Okay, now we are going to focus on this endomembranous systems, that is endoplasmic reticulum. So endoplasmic reticulum is actually an endomembranous systems, an extension of the nuclear envelope, and then it has been thrown into numerous folds. Again, I'm not going to detail because you guys may be knowing a whole lot of this one during your class 12s and also during your preparations for this AIIMS exam. So just remember that these are thrown into numerous folds, and the whole purpose of putting them into folds is to increase the surface area. On the top of this endoplasmic reticulum, there is something called the ribosomes, which actually sits over there. Those are the factories where the proteins are actually being synthesized. So this ribosome synthesizes the protein synthesized enters into the endoplasmic reticulum. From their vesicles buds, and then they goes to the Golgi complexes, and from the Golgi they actually travels further. Now this part of this entire thing is known as the endomembranous systems. So this is a wonderful system of communications, something like an highway, roadways in highways, where you see that the different levels of this. There is in check posts are there, the toll bridges are there, where individual things are actually been monitored and then they actually proceed. So that's exactly what is happening over here. This is the endoplasmic reticulum. A little more detail about this one, if you had to actually separate out in a schematic diagram. And this is the red dots over the surface of this one. This is actually the ribosomes which is sitting on this endoplasmic reticulum, making it as a rough endoplasmic reticulum. If you have something called the rough endoplasmic reticulum, there is something called the smooth endoplasmic reticulum too. And those smooth endoplasmic reticulums are actually the sites for synthesis of steroid hormones, right? They are different from the proteins which are actually produced in the smooth endoplasmic reticulum. We are going to come on that also later on. Okay. I have a small video. I don't know whether this is going to work or not, but I'm just trying to, make it work. If not, we are going to, proceed with that one. So anyway, this is the URL for the video. It seems it doesn't work. So we kind of shifted to the next slides. So this is the secretory model or the secretory pathway within the cells. Again, as I said, that this is something like the highway system where the proteins are synthesized, and then from there the vesicles bud off from the endoplasmic reticulum, then they travels all the way to the Golgi complexes and actually exits through the plasma membrane if the proteins has to be secreted out. Otherwise they are going to be transported or transferred to any one of the organelles within the cell. Okay, so that's the route in which it actually follows. Again, everything is actually written at the corner of that slide. So feel free to go through it and you come back to me if you have any queries. I mean, this is very easy. I mean, you are brilliant guys. I mean, you don't need so much of explanation to tell all those simple things. So when someone cracks AIIMS exam, we say that they are the best of the best in the India. So, these are very trivial for you people. So just, even then, if someone of you isolated one or two who could not follow clearly because it may not be audible or it may not be clear, then feel free to ask me. Okay. So that's the nucleus, and this is how the nucleus looks like if you take a section of it. And if you look under electron microscope, remember a few minutes back I was talking about electron microscopy, and then this is how the nucleus looks like. And this is when you magnify them, then you actually looks them something like that. Now these are called the nuclear pore complex. This nuclear pore complex are the gates within the nucleus which actually allows the substance to come out of the nucleus or enter within the nucleus. Again, someone is going to talk a detail about this. Even I am going to talk about this one, nuclear export and import, in the subsequent classes. But these are the gateways within the nucleus that allow the substance to go into the nucleus. Entry into the nucleus is not a free flow. It's not something anyone can go inside it. It has to be highly regulated process. And this is actually been brought about by these nuclear pore complexes, which are themselves an aggregation of the proteins that actually forms the gate and allows only a selective substances to enter through those gates. Okay. That is the nuclear pore complex. Okay. So nucleus contains the genetic informations. All of you know it contains DNA, RNA. Then nucleus is been covered by nuclear envelopes, and we have the nuclear pores. So I was talking a little back, openings which allows the movement of the molecules in and out of the nucleuses, and then it contains DNA, and then as we are going into DNA, we are going to the separate domains. So we are not talking about genomics over here. This is still cell biology, so we are going to restrict ourselves and not. It's very tempting to go into DNA. We are not going to it. We are going to kind of control our temptations and then actually just talk about cell biology today. So this is the endoplasmic reticulum. We talked about it. Now just want to focus on you that there is something called the rough and there is something called the smooth endoplasmic reticulum, which does not have the ribosomes. Apart from that, they are just exactly the same, but the functions are different. Though they don't have ribosomes, they are mostly important for synthesis of the lipids, synthesis of the steroid hormones, synthesis and detoxification of the chemical wastes within the cells. So that is another place where the smooth endoplasmic reticulum detoxifies the drugs and the other kind of toxic products within the cells. So they had a different sets of functions. Okay. So rough endoplasmic reticulum, this is just the summarizing of this one. Again, I'm going to go a little fast. I'm just going to focus on the smooth one: fatty acid elongation, desaturations, steroid biosynthesis, glycogen metabolisms. Those are the domains of this smooth endoplasmic reticulum. What is fatty acid elongation, desaturation? When you're going to study about fatty acid biosynthesis, is going to be a 1 hour worth of lecture where someone is going to talk that how fatty acids actually been synthesized from acetyl CoA residues, and eventually after these fatty acids are actually been assembled from those monomers, the big chain fatty acids has to be undergone various kind of chemical modifications known as elongations and desaturations and saturations, where you kind of put the double bonds and all this kind of wonderful chemistry over there. Those things still happens in the smooth endoplasmic reticulum. Again, I am not going into that. We are going to keep it for the next classes. Now, last one is detoxification. That I was talking a little back, that the smooth endoplasmic reticulum is also engaged in the detoxifications. Now, another important component of the cells after endomembranous component is the Golgi complexes. Now, this is how the Golgi complex actually looks, and the endomembranous structures looks. Again, under electron microscope, you could see that is thrown into numerous number of folds, and all of these folds are actually just an way to increase the surface area so that more chemical reactions can take place. So it's all about maximizing your capacity within the small cell so that you could actually maximize the chemical reactions, and this is done with the help of this throwing them into large number of folded structures. So wonderful topology over there. Again, very interesting. Another thing I just wanted to talk over here, just the two jargons. There is something called the cis and there is something called the trans. So the Golgi complexes which is closer to the nucleus are known as the cis direction, so the cis phase, and the Golgi complexes which are farther away from the nucleus near the plasma membrane are known as the trans one. This is the trans phase. This is just about the name, nothing spectacular about them. This is just about how we actually tell about the vectorial directions from the cis to trans, right? That's how the movement actually takes place. Okay, so nuclear transport, we already discussed about that. We also already discussed about this one. Now we are talking about ribosomes. These are the factories for the protein synthesis. All of you know them. This is very fundamental. We study them in the basic biology, so we are actually going quickly through them, and we are not going into any detail into the different components and the subunits of the ribosomes, right? They are the different components and the subunits of the ribosomes, you know for sure. I'm not going to talk about them, the 60s subunits, the 40s subunits. So those are again part of this discussions when someone is going to take classes on protein synthesis. I'm going to leave for those people. We are just going to go into a wonderful event that happened in 1906 where someone was awarded the Nobel Prize for medicine because of the discovery of the Golgi complexes. So these are again part of the endomembranous structures where people found that these have been associated with something called the glycosylation reaction. So once the proteins are actually been synthesized by the ribosomes sitting on the endoplasmic reticulum, then these proteins have further been modified, and this modification is important for the proteins for their functions. Remember in the first phase I told you in one of the slides when I was talking about plasma membranes is that these glycoproteins are very important components of the proteins. Without these glycoproteins, many of the proteins are not going to function. Now, how the proteins gets glycosylated? This is this guy who is doing this function. This Golgi complexes. What they do is that they have a series of complex factory of enzymes, and with those enzymes they actually add on the sugar residues to the proteins and forms the glycoproteins. Again, glycoprotein is another separate topic of a discussion, so we are not going to that. And this is how the Golgi complex actually looks under the microscope and electron microscope. So again, it's thrown into large number of folded structures. They are all parallelly stacked with respect to each others. Remember, we have the Golgi complexes. The plants also have the Golgi complexes, and we call them as dictosomes. So dictosomes are actually the Golgi complex equivalents of the plants. Okay. So those are again the same structures, glycosylations, modifications of the proteins. There is something called the post-translational modification, when the protein is actually been synthesized, and after synthesis of the proteins are actually been added on with the sugar residues to form an additional functions of the proteins, like the blood group antigens, like the receptors on the cell surface. If they are not being glycosylated, then the proteins won't function properly. So that's what this guy is actually doing, the Golgi complex, efficiently glycosylating the proteins so that they can restore the biological functions. Just in making the protein is not important. They has to be also given the biological functions, and that's what the glycosylation actually does. Now this is the route which I was talking back. Again, we shown this slide, so we are just quickly going through it, and we are not focused too much on that. Now this is something which is the lysosome. This is also known as the suicidal bag of the cells, where it contains whole lot of these hydrolytic enzymes. But then lysosomes is something which is, you know, an innocent creature. I mean, I just put it in a satiric way, that this is an innocent creature which is unnecessarily been, you know, kind of given a bad name. They say the lysosomes contains all the hydrolytic enzymes. Its whole function is to digest the substances. That's true. There's no debate on that. But at the same time, it is also true that if the lysosomes does not perform the functions efficiently, then you have a plethora of clinical conditions. I'm going to talk about them, couple of them, which are going to come in if the lysosomes does not function properly. So these lysosomes are actually the garbage cleaners within the cells. They kind of kill and destroy all the dead and the old and the weird out the proteins and the organelles. In this electron microscopy image, you could see a lysosome which has already engulfed a part of this mitochondria and also a part of another organelle, and therefore it is in the process of constantly eating not only the proteins which has to be degraded, but also the organelles within the cells. And over here I'm going to use a term that is known as autophagy. So autophagy is a way where the cells actually eats up its own. So this lysosomes are actually autophagous in its nature, is that they engulf these different cellular organelles which are worn out and which are old and which are non-functional, so that in the cells we don't accumulate garbages. This is very important. If the garbage accumulates in the cell, we are going to have a serious problem. I'm going to talk about eye cell disease, and I'm going to talk about those serious problems right away in a couple of slides. But this is mitochondria. The mitochondria is a fantastic part of the cells, right? It's a part of the cells, but really not a part of the cells about 4.5 billion years back when the life was evolved. Life evolved 4.5 billion years back, right? And that is about 750 million years after Earth was actually been born. So after Earth was born, 750 million years we waited, then the first life came, which is about 4.5 billion years back. And after 4.5 billion years back, you know, when the first life actually came into existence, then pretty much that was actually an anaerobic life. Now something like about 2.3 billion years back, we have the first oxygen producing creatures that actually starts evolving, which is about 2.3 billion years back. And what has been found, that if you have an oxygen, then the chemical reactions, now it is pure chemistry, but the chemical reactions can be further enhanced, the chemical reaction capacity can be further enhanced, and therefore cells has actually started, you know, looking for a way to increase the oxygen consumptions and to utilize the oxygens. And at that time, there possibly this is an endosymbiotic theory which is actually making a story out of it, that at that time what happens is that possibly a bacteria has the capacity to do that, and what the cell did is that they engulfed those bacteria, and those bacteria now became a part of the cell. So this bacteria has now become a part of the cell, and now the cell who has engulfed this bacteria got a survival advantage. So everything is within the Darwinian theory of evolution, because if the cell does not absorb this bacteria, it's not going to survive properly and efficiently. But those cells which have successfully engulfed those bacteria, which can produce oxygens and which can utilize oxygen, then they found that their efficiency has been tremendously enhanced. And that's how those lineages of those cells actually got an survival fitness in the evolutionary landscape. And eventually we have those eukaryotic cells, today's eukaryotic cells, which has this mitochondria, and this mitochondria is actually has its own genome. We call it the mitochondrial DNA, which is different from the nuclear genome, which is known as the nuclear DNA. And this mitochondrial DNA actually encodes protein for the mitochondrial functions. So mitochondria is an organism within itself, but at the same time it's a part of the cell, and it's the whole function. One of the function of the mitochondria is production of the energy. But then if you just say it's the powerhouse of the cell, that's something like downsizing the entire function of the mitochondria, because it has so many different functions that we need about 2 classes to, you know, just enlist the function of the mitochondria. But it's been related to cell proliferations, apoptosis, autophagy, epigenetic modifications of the cells. You may not know what is epigenetic right now, but mitochondria actually does whole lot of different interesting functions other than producing ATP from chemical reactions. So it is just not the cellular energy factory, but it is also a hub of so many different events which is happening within the mitochondria. So that's the mitochondria is, and then these are the chemical reactions within the mitochondria. Just see what important chemical reactions are. TC cycle, urea cycles, heme biosynthesis, fatty acid oxidations or beta oxidations, amino acid metabolisms, gluconeogenesis, synthesis of organelles, proteins. Now without this TC cycles, you cannot have the ATP which can go into the, which can be utilized. And this mitochondria has the electron transport chain which utilizes this ATP produced, which utilizes this electron gradient and produces the ATP. And then you have the urea cycle. Without the mitochondria, you cannot have the urea cycle. And then the ammonia is going to build up. And you know that ammonia is potentially very toxic. It's going to immediately affect the central nervous system. So we need this ammonia to be detoxified to form the urea and excreted out through the urine. Again, mitochondria comes into savior. Then heme biosynthesis, fatty acid biosynthesis. Each and every one of these are actually topics of your curriculum, and you're going to study a whole lecture on that. So you're just going to get an overview that how crucial the mitochondria is. Again, this is something called the structural components of the cells, the one that actually gives the cell the shape, the capacity for the cells to move, and the capacity of the cells to undergo migrations. So this is known as the cytoskeleton components of the cells. Those are so many numbers. It's everything is there in the slide. We have the microtubules, microtubular structures. We have the centrioles. What is the function of the centrioles? Again, most of you are aware of this one. One of the main function of the centrioles is to cause the movement of these daughter chromatids from one pole to another pole, which is actually been shown over here very elegantly in this schematic diagram. That what the centrioles are actually doing is that they are part of these microtubular structures where they help in integrating the movement of this chromatids from one end to another end, so that they can be equally distributed between the two daughter cells. Fundamentally very important. If you don't have this one, we are not going to have the cell divisions, or if it happens aberrantly, then you are going to land up in cancer. That's exactly what happens in cancer, is that we have something called ploidy, where the cell has more number of chromosomes than the 46 chromosomes. And why it is like something like that? The reason is that there is an unequal distribution of these chromosomes. If particular cells will have a lot of chromosomes than what is actually required in the normal conditions. Again, these are the problems of these cytoskeleton components, centrioles and microtubules and so forth. So this is microtubules, and these are the cilius and flagellus, which is part of mostly prokaryotic systems, but then we found without a reasonable doubt that there is lots of cilius and flagellus even in the eukaryotic systems, in the airways, in the intestinal brush borders. These cilius and flagellus are so important in the reproductive tract that if they are not there, the female is not going to conceive and is going to come with the reproductive failures. So these cilius and flagellus are very fine finger-like projections, like brushes, and they help in the very gentle movement, not very harsh but very gentle movement, so that gradients of chemokines, gradients of cytokines, gradients of hormones can actually be generated. And as a result of this gradient, we have different biological function. Think about this. We just have one chemical. How can you have multiple response from this one chemical? One way to do that is that you create a concentration gradient of that one single component. And how do you do that? With the help of the cilius and flagellus as one of the components. They swipe the chemicals in a way that one side has a lot of concentrations, and slowly there is an exponential decrease, and you have an entire spectrum of the concentrations. Each of the spectrum of the concentrations is going to start a particular kind of a chemical reactions. So these cilius and flagellus are absolutely important for so many different functions, and this is proved beyond a doubt that they are important for the normal cellular physiological functions. This is then confocal macroscopy that shows the wonderful fiber structures within the cells. This is something like an architecture of the cells. If you go into a building which has not yet been handed over to the residents for staying, then you will see that all of this within these walls of these buildings, we have these crisscrosses of these iron rods, right, which form the framework of them. That's the framework of the cells by the cytoskeleton components of this one. And then they also help in the contractions. Again, I'm not going to talk about this. The people in physiology is going to spend possibly weeks and months talking about muscle movements. Again, we are skipping this one. This is how the cell actually moves, the amoeboid movements and so forth. Again, this is possible only with the help of the cytoskeleton components. Very important, very, very crucial for the cellular movements. Remember, cellular migration is fundamental for so many important things. If they have an inflammation over here, the neutrophil has literally to move to this particular site. How it is possible that the neutrophils and the other immune cells can move? It is with the help of the cytoskeleton components, then they can actually show this locomotion. So this is also a very important fundamental component of the cell. And look how they looks under microscope. This is known as confocal microscopy. We put an filament, this is actin filaments, we put an antibody to it and stain them, and this is how fantastic they looks like. The nucleus is blue in color, the cytoplasm is reddish in its nature, and then we have these green fibers of this actin, because you have used an antibody against actin and then actually did an confocal microscopy. Again, detail you are going to study in instrumentation classes, where you could see that the different stress fibers within the cell. So it's a very elegant structures. I mean, if you look under microscope, you'll be amazed to see the beautiful architecture of this nature. Okay, so we are going into something quickly. This is one slide which I wanted to show you, just to kind of focus on today's class in a more realistic sense, that what are the implications of this one. What is this one? This is a condition that is known as eye cell disease. I for India, that eye cell disease. Now what is that eye cell disease? Eye cell disease is also known as mucolipidosis. To be very precise, this is mucolipidosis type 2. What is that mucolipidosis type 2? This is an lysosomal storage disorders, where the lysosomal enzymes are not been functioning properly, and as a result of that, mucopolysaccharides actually accumulates within the cells, to that extent that it completely fills the cell with this garbage of these mucopolysaccharides, and ultimately the cells actually goes for cell death. Now imagine that is happening to each and individual cells in the body. Is going to be devastating, right? Now these individuals with mucopolysaccharide, with the eye cell disease or mucolipidosis type 2, they are going to have coarse facial features, skeleton deformities, abnormal growth, and skeleton deformities, abnormal growth, and absolutely poor quality of life. They are not going to survive even second or third decade of the life. So it's very fatal conditions, just because a few enzymes in the lysosomes are non-functional and they cannot degrade. Their function is to degrade those mucopolysaccharides, but they cannot do it properly, and therefore the things gets accumulated, as if the garbage has not been cleaned from the house, and the things gets accumulated and produces a filthy environment within this, and then it actually ends up with this particular disease. So now you understand that cellular components are so crucial, so important, because if they are disrupted, they are going to have serious consequences. This is something called the MELAS, which actually stands for mitochondrial encephalopathies, lactic acidosis, and stroke-like episodes. Again, this is because of the mutations of the few genes within the mitochondria, because of which the mitochondrial function is actually been compromised, and then this individuals goes into frequent episodes of seizures and fits, and again this is a very fatal conditions. So again, this is because of the mitochondrial defect. Now this kind of tells us that you take each and every organelles within the cells and then understand that how important those organelles are. One way to know that is that disrupt the function of the organelles, and then see that what is the disruption in the cellular physiology, and you will see that there is a tremendous change in the cellular physiology, which shows that each and every organelle can cause a fatal disease, and therefore these organelles are so crucial for the survival of the cell and for the survival of the entire organisms, right? Now we have come to the end of the class. I am just going to summarize with just 4 points. The eukaryotic cells has evolved to contain large number of organelles. Many of the organelles are actually enclosed within the membranous structures. Again, I told you why, because we need to maintain the privacy of those different organelles, so that each organelle does not disrupt the function of the other organelle, but at the same time there is a communication channel between these two, or between the different organelles, and then they function as in a very nicely orchestrated manner. And this organelle actually performs dedicated functions. That exactly I was talking just right now. And disruption of this process can lead to cellular pathology, and that's exactly I was talking about just now, about this lysosomal storage disorders. There is something called the lipid storage disorders. Again, there is Niemann-Pick's disease. There is so many different kinds of lipid storage disorders are there, which you are going to study detail about in your subsequent classes. I don't want to take the credit out of those classes, because there are so many different pathologies associated with these lipid storage disorders. As I mentioned, Niemann-Pick's disease is one of them, where we have these conditions where the abnormal metabolites and the abnormal components and the chemicals actually accumulates, because the lysosomes cannot degrade them, and eventually it actually leads to the formation of these inclusion bodies, and then compromises the cellular functions. There is another something called the Tay-Sachs disease, which is another mucopolysaccharide disease, where there is no storage disorders, and again all of these G1 gangliosidosis, which is another one where the gangliosides actually accumulates, and if they accumulate in the brain, then the function of the brain is actually being compromised. So most of these individuals are mentally retarded. They may be about 12 or 15 years of age, but their mental IQs is just like 2 years of a child, because of the reason that there is an abnormal function of this central nervous system and the brain development is completely been rudimentary. So having said that, we understand, we appreciate that the subcellular components of the cells are very crucial, very important biological functions associated with them, and if those are disrupted, then we are going to land up with different cellular pathologies. And then we talked about something called the cytopathology, where the cytopathologist looks under the microscope and see the abnormalities within the cell, within the tissue, within a tissue block, and then tell that okay, this cell has this abnormality and therefore it might be associated with this disease. That's a fantastic thing out of this. Now I'm going to show you a slide where you're going to study a couple of books. Again, no hard and fast, but a basic. It's good to have a list of the books. You could, each one of you can be comfortable with one particular book. You don't have to purchase all of them, but each one of you may have the different comfort levels. So you could actually, kind of purchase one of the books which fits your comfort level. Go to those books, and there are a couple of chapters which are free. You could just scan through them and then see that what is the language, in what level they are actually talking about, and then you could actually purchase the book. You could hire it from your seniors. So Harper's Biochemistry, which was talked about earlier, Leringer's Principle of Biochemistry, this is a little more exhaustive. So for the people among you who are a little more interested in knowing the detail about biochemistry, this Leringer is a fantastic book which talks in a very great detail about the biochemistry in a story kind of telling. Then you have the biochemistry book by Stryer, very good, specifically the chapters on hemoglobin and oxygen carrying capacity of the hemoglobin. Biochemistry by the Stryer is fantastic book. There is something which is not listed over there, which is White and White, VOET. There are two whites over there. Those White and White book is fantastic for enzyme kinetics. So someone wants to study enzyme kinetics, just don't study anything, just go to the White and White in the library, just read that chapters on this book, and then you are done with that. They are the best in enzyme kinetics. Now do we have the Devlin's book of biochemistry, a general overview of this biochemistry, pretty good book. And then we have the book by Professor G. Pitala, who used to be our AIIMS, head of the department of the biochemistry department where I work in, and he was there for large number of years, and then he has actually written a fantastic book on AIIMS on biochemistry. So again, you can actually go and read his book. He's an immunologist, but he actually written a fantastic book on biochemistry. And then we have the Treat's book on clinical biochemistry, which is again very important book for clinical biochemistry, most of the things which we are going to do in a practical exams. And then we have the Lippincott biochemistry, which is a very book with a lot of illustrations, fancy pictures, images, very good to understand and very quick to grasp, similar to that of the Harper, but then the presentation is different. There are a lot of MCQs and SCQs questions at the end of them. So fantastic books. Again, you could actually read about them and you will have an overview of them. So Lippincott, I'll suggest that it's kind of more for kind of quickly summarizing the whole thing, but to start with the concepts, it's better that you start with the Harper's and the Stryer's and Devlin's book. But again, this is a personal choice. I leave it up to you. Whatever you're comfortable, which fits into your comfort zone, go for that one. So with that, actually, I end today's class, and you could switch on your microphones and bombard me with the questions. If you have anything, I'll be happy to take your questions before I can kind of leave, and the next group of people are going to come and talk with you. So any questions, anything, please feel free to talk to me right now.
So there is no harm in buying all of them, right?
Tell again, a little louder.
There is no harm in buying all of them, right?
No, nothing harm. If you have everything, only one harm is that your friends will be bothering you. They will asking you to lend the book, this and that. So I was just kidding. So there is no harm in purchasing all the books. So have them, and each book comes with the different flavors in the different ways of presentations. And a few of the chapters, as I mentioned, are very good in a couple of books. So if you don't want to invest, we have a fantastic library with a huge volume of books. So you can actually go over there and actually consult it. So did I answer your question?
Yes, sir.
Okay. Anyone else? Anything about today's class? Anything else you want to talk about? About COVID-19? Anything?
Well, the vesicles which are coming out from the trans space can go back to the cell space or endoplasmic reticulum.
We have something called we have we have something called the anterograde and the retrograde movements. Yeah, those kind of things are possible, where the things can come out from the cis, goes to the trans, from the trans it can actually go back to the cis. So this kind of shuttling is always possible. Yes. Anything else? Anything related to biochemistry, Percy? Because today's class we are kind of independent because nothing is started. So if you have a lot of curiosity, feel free to ask. I don't know if I can answer all of them, but I will try my best to answer all of them.
Sir?
Yes, please.
Sir, which book we should consider to buy finally?
I would say that if I have just, you know, the option to purchase only one book, if my dad gives me the permission to just purchase one book, I'll go and purchase Harper's Biochemistry. The reason being is that it's kind of, you know, assimilations of everything in a pretty good way. So Harper's Biochemistry is the book which I would say that if you want to book one book, go for Harper. If you want to purchase two books, then maybe Harper and Strayer is okay. So again, you know, if you are kind of keen to study multiple books, like someone has asked, you know, our library has all of these books, so you know you can always consult and, you know, come and look into those books, go through a couple of chapters, and then whatever if you think that that book is within your comfort zone, you can actually go ahead and purchase it.
Thank you, sir.
Okay. Anything else? So in our next class, we are going to talk about membranes, the biological membranes, and we are also going to talk about the transport across the membranes. Very important. It's not important because I am taking the class. I mean, if it had been anyone else, I would also still vouch to the same thing, that it is very important, because, you know, the membrane is the first structure, the largest structure, and then a lot of things actually happens at the level of membranes, and this membrane biology, transport across the membranes, the active transport, the passive transport, the diffusions, the facilitated diffusions, the whole lot of things actually happens within the membranes. The cholesterol, their role into the membrane biology. So we have so much interesting things happening in the membrane, and so much different pathology associated with the membrane's dysfunctions, that it is a chapter, it's a book which, or it's something like an individual lecture within itself. So we are going to talk about membranes in our subsequent classes. Is your class time over? Okay, anything else, please feel free to ask.
Sir?
Yes. Yes, please. Yes.
Can you elaborate how we have to study now?
Tell again a little loud.
How we have to study now?
How you have to study now? I mean, you are asking that how to study this back? Yeah. Make your.
I mean, I'm going to be used to study and time it. So what is the way of study now?
Yeah, so the way of study for every lecture is that, you know, we are going to share you the PPTs. So what I recommend is that it might be a little annoying, but it is best that you invest your time immediately within the same day when the class has been taken, because, you know, our memory is very short lived and you have hundred of things bombarded in your mind every single moment, so you tend to forget this lecture and the important finer points. So it is annoying, and at the same sometimes it is even more demanding, but that works fantastic, that, you know, just after the class, within that day, within the 24 hours. So what research shows that within the 6 to 8 hours we start losing our memory very fast. So within this first 6 hours after the lecture, 6 to 8 hours, at the max 12 hours, you know, read about, go through the slides vigorously and then know everything. Then that actually adds a lot of volume to your understanding, because if you do the same exercise a week later, you might have to work 2x or 10x more to understand and recapture all those points. So go back, read and know about the slides today itself, so that you can maximize the class, and eventually you go back to the book and then see that what I had actually left out from this one. The reason being is that not everything is possible to be covered within a 45 minutes class. Neither it is, you know, desirable to do that one, because then we are going to, you know, make this class very boring and lethargic. So we kind of give you the key points and the key important discussions, but it's your duty to go back to those chapters, to the PPTs, and then go back to the chapters and read them into more detail, because you have wonderful illustrations and then images, pictures, case discussions, all these things happening in the book. So book is the ultimate friend for you. We are just kind of giving you an overview and a bird's eye view about the things. So go back to the book and then finish it off. After that, if you have a problem, shoot us with an email, WhatsApp, SMS, phone call, whatever the way you could do it, and ask us the question that this portion of you could not follow, or this thing you covered it very fast, we want it to be taken second time, or this thing is not, the image is very nasty, it's very blurred, give us a different image of the same thing. So we'll be happy to kind of go back to those individual demands and then actually respond to you. So for now, I think it is best is that immediately go through those PPTs and come up with your understanding about what has been taken in today's class. Did I answer your question?
Yeah, thank you.
So someone if you have asked that, sir, is it required to make notes by heart? So what I would say is that note taking or taking a note is very important. Even though Percy in his electronic medias, I mean everything just your Android is there, you don't need anything to be done. But having said that, taking a note is a very good habit, because eventually when you're going to sit in the clinics, you see that this particular habit of you is going to make a difference like day and night. Because when you take the notes, what happens is that you carefully jot down the points, and those points gets embedded in your mind, and then that helps you to formulate the entire thing very nicely. So taking a note itself is a very good practice, is a very good behavioral practice. Apart from that, it may be a little difficult, because you may be tempted to use your iPhone or iPad or some kind of a thing to quickly scan the whole things or take a snap of the PPTs or whatever it is. But taking a note is the ultimate thing. If you could do it, at least the key points of the class, that's going to suffice. You didn't have to take word by word and point by point, just the important fundamental point what is important, and then that's going to suffice. Okay, so that answers the question. So which book is good for cytology? I would say no, don't consult any book for cytology. That is a very focused domain of pathology. So you are going to learn either a pathology in your second or third year. So don't go into cytology right now. You have enough food on your plate. And once you come back over here, you have anatomy, physio, biochemistry, practicals, dissections, so many things. So I would not recommend right now doing great into cytology. Just overview about that is important, because that is something either we are going to learn in a pathology lecture series, which is going to start possibly in the next year or later on. So WhatsApp number, yes, our senior residents is going to share you with the WhatsApp number, Maya and all the faculties, and I'm also going to give her the PPT. She is going to send the PPT to the monitor of your class or to the WhatsApp group, so that by next an hour from now, all of you should have the PPT of this class today. In this PPT, if you can't follow anything, feel free to ask me back. Okay? Anything else? The other thing what I would say is that biochemistry is a pretty volatile subject. So try to have every day a little bit of biochemistry, kind of, even if it means that 10 minutes, go for this one at least 10 minutes, because it's kind of very volatile subject, so you kind of forget it very soon. But having said that, biochemistry is also very fundamental in your understanding and correlating a lot of other things. Like if you go to physiology, you will see many of the things you could correlate very nicely if you have a fundamental basis of biochemistry. So it's very important that this core STEM subjects, those, I would say STEM is not the real right word, but still then this biochemistry, anatomy, physiology, whatever the preclinical is been taught in your first year, those are very important. They are the foundations of your understanding in the later years. Because if these are actually kind of on a shaky ground, then you may not be very comfortable once you go into the second year. So try to be as much as sincere as possible in the first year, trying to master these subjects, because you are not going to come back to the biochemistry, anatomy or physio later on, right? You are going to go into the next curriculum, but eventually these are going to be the building blocks for your future knowledge, that's for sure. Okay, if none of you have any questions, so I am taking a break and then handing over to the next people who are going to carry it forward from there. Okay, so wonderful. Have a nice day. Stay safe. Wear mask. Wash your hands with soap and stay safe from coronavirus. Okay? Wonderful. Thank you.
না না আপনার ওখানে নেটওয়ার্ক ধরতে পারবে না আলাদা নেটওয়ার্ক কিন্তু এটা কখনো বন্ধ হবে না এটা কখনো বন্ধ হবে না আর এর হলো ব্যাটারি থেকে একটু বড় আছে আমাদের তাই লোক কোথায় ওরা তো নেই
হ্যাঁ Hold on for one moment. I just called the residents, so hold on for one moment. Let me see. What to do?
All good. I'm done. I got it. All can be done online.
Yeah, you took that in the class now. I have only 1 hour class. How? After that they can go.
They can go. They should just come back at 2 o'clock.
Oh. Hello. Hello. Hello.
Yes, sir.
Hello. Yeah, you people are going to come back at 2 o'clock. Okay, 2 p.m.
Yes, sir. Okay, sir.
Please send the message in a WhatsApp group so that everyone is aware of.
Yeah, sir. Everybody knows that.
Okay, wonderful. Thank you.
Thank you, sir. Thank you, sir.
Siddharth, this is very
difficult. No?
I could not see any slides.
I know. But luckily from now on we can have it anywhere else. We don't have to come here.
Anyway, is it?
Yeah. I asked him. He'll send us that link, that online link, and we connect through that only.
But our home computers will go home bandwidth and sustain this. How long is it out? I can switch off
You can just log out.
I'll switch off this thing, projector. That's cancel. Let me cancel the meeting first. What is this written?
Full screen mode, your notifications.
No.
Required.
Yeah, so
I think we will have to do this only online.
okay, yes, that.
This is too difficult.
Yeah.
Coming over here is too difficult for a lecture.
Yeah, I know.
It's better to do it
from your home.
Yeah, home office or somewhere else.
Yeah.
Here there's so many things to coordinate.
So many things, and then you have to see, and that goes covered.
No, I think. See, I've spoken with everybody. It can be done in hybrid mode.
Wonderful.
Okay. So this is the letter, which is you've seen it already. Okay.
But this is your side.
So this is the specs, and this is for the thin plan. Okay. So that's all.
Now we are asking for only 11.
9th only.
Oh, 9th only.
9th. So because everybody is free on 9th. The only thing was whether or not we should increase. So I had already taken dates with everybody.
So 9th, where No, why are we going to?
Where are we going to be? You tell me where are you going to
No,
be?
is it everyone will be online or what?
So now that we have to, because we have not said, we said 250 over here. And
I think we definitely have to.
We can be there.
Generate and Google it.
That we will do.
For the people otherwise,
we can meet on
what is 9th? 9th is
Tuesday, Wednesday.
Wednesday. Okay, then Wednesday, at least we will
be there. Two of us will be there.
But inform to the sir.
I'm sending the letter now.
Okay.
He said don't hold the meeting on the 4th.
Okay.
That's all.
4th? Previously. Okay. Now he's okay with it.
Now he's okay. So we'll send this. You want to check it?
No, that's fine.
Everything.
We already sent it. That's one thing. Okay.
So then 215 on that day.
Okay, 250. Which one you are talking about? Today's one?
No.
On that? Yeah, that day. I'll be there for the whole day. Today, why at 2 o'clock? Why are you going to come?
Where am I going?
Yeah, in this one.
We have nothing, no?
It's just
practicals and SR.
So SR will manage.
We have nothing to do with that.
ठीक है.
So then I give this to the office then?
Give it to the office. Okay, sure. Hello. Vincent. So I asked them to come at 2 o'clock. Is that okay? Okay. Why we will be there in 2 o'clock? Oh, you will come over here in LT only. Okay. Okay, that's fine. I told the students and they are also aware of that.